experimental therapeutics. It is truly heartening to see such academic spirit and eagerness in the hall this morning. As we move ahead with our sessions today, we continue to explore some of the most impactful developments in the world of drug discovery and molecular therapeutics. I would like to express our sincere thanks to the Ministry of Education, Government of India and Panjab University for supporting global initiative of academic networks, which brings globally renowned scholars and cutting-edge topics to our doorstep. Now I would like to welcome Dr. Aman Bhalla for the tutorial.
A very good morning to all present. Okay. So I just give a brief glimpse that what we have done and now what is the objective of today's lecture. So we have started with the need of the asymmetric synthesis. I guess you have a clue about that why we have to study about asymmetric synthesis. Once you know all about the initial definitions and the concept, then we move to the methods which are required for the synthesis. Okay. So the methods which we discussed yesterday, that is, one is the resolution, second one is from the chiral pool, third one is the enzymatic. Okay. So now the fourth one, which is the chemical synthesis starting from the achiral substrates. Okay. I guess you have a keywords with you. Clear? So for this fourth, that is the asymmetric synthesis using the achiral substrate, we have to study 4 generations. Okay. That generations which will tell you that how you are going to plan the asymmetric synthesis. Yesterday I showed you one reaction, that is asymmetric aldol reaction. How many number of products are possible in that? Can you just give me just number? 4. Very good. That is 4 products will be there. And what will be your objective and target as per the requirement? Okay. What is the requirement? That you want to attain all the 4 products in a pure and enantiomeric form. If any reaction planned and it is giving you exclusively 1 product out of 4, what will you categorize that reaction? It is enantioselective or enantiospecific? Enantiospecific. But if there is the percentage in that 4 molecule about the majority and minority, then we have to categorize that reaction as enantioselective. Okay. So these concepts already with you because they are required to study these generations, that how a asymmetric reaction can be performed. Okay. So asymmetric synthesis is possible only under the influence of chiral group or chiral auxiliary. They may be derived from the chiral pool, that is okay, but if you have to carry out asymmetric synthesis, chiral unit is required, because if there is no chiral unit, then it is a 2 dimension. Because in start, if you see my first slide, I shown that is chirality, that is 3 dimension in 3-dimensional plane. What is the arrangement? Okay. So depending upon how this influence can be exerted, 4 strategies have been developed for the asymmetric synthesis. Okay. Today we will only follow these strategies. So see very carefully. For all the asymmetric reaction, whosoever is preparing for CSIR, NET, UGC GATE examination, this is the important component of asymmetric synthesis. Now your first generation is always related to substrate controlled. Okay. So I just say you have to remember only the keywords. I am not saying that you have to see the mechanism aspect, you have to just remember keywords, then it will be easy to understand. Okay. First generation, that is substrate controlled. Okay. Now what is that? The chiral environment is present on the substrate. Can you see in your starting substrate any chiral unit? You have a CH3 and hydrogen. The CH3 is present above the plane and hydrogen is present below the plane. The chiral environment is present on the substrate and the formation of new stereogenic unit. Can you see in your product you got some new stereogenic unit where you have a methyl and OH. OH is above the plane and methyl is below the plane. So the chiral environment is present on the substrate and the formation of new stereogenic center is controlled by this. Now see, if you have to perform a reaction with cyclohexanone, what is cyclohexanone? 6-membered ring with carbonyl unit. As we discussed earlier, carbonyl unit is a planar sp2. So if you have to introduce any group on the carbonyl carbon, you have a possibility of 50-50%, either from the above the plane or below the plane. So in your starting substrate you have already placed one chiral unit adjacent to the carbonyl group. Okay. Now you react it with methyl magnesium iodide or we call it RMgX. तो आपने एल्डिहाइड एंड कीटोन के रिएक्शन पढ़े हुए हैं RMgX में. If you react with formaldehyde you get primary alcohols. If you react with acetaldehyde you get secondary alcohols, or if you react with ketone you will get tertiary alcohol. So this reaction is very simple. क्या रिएक्शन होता है ग्रिग्नार्ड रिएजेंट के साथ कार्बोनाइल का? How this reaction is going to pursue? Have you checked the reaction? If I just write you are reacting with methyl magnesium X, okay, Grignard reagents. ये marker check कर देना. Now what is the reaction? What is the simple chemistry in this reaction? You say that this is your partial negative and this is your partial positive. You are bringing this negative to this carbonyl carbon and this polarity will drift to O negative. So your methyl is going to attack at the carbonyl carbon. This is the simple reaction of aldehyde and ketones. Okay. So when we are talking about the movement of this methyl group to this carbonyl carbon, we are moving it this. What will you get? RCO negative, methyl and H. ये जो MgX है, it will come to here, you will have this. Then you treat with water, you will get this will go out. This is the simple reaction which we have studied in our +1 and +2. Now the question is a very simple question. When you are seeing this methyl group coming to carbon carbonyl, you are seeing it in 2 dimension. Your blackboard is 2 dimension, but actually you have to imagine this carbonyl group is in the plane. When a methyl is coming, it can come from the upper side and it can come from the lower side. So the possibility is 50-50. But if you get a 50-50 both the enantiomer, that is your racemic product. Then what is asymmetric synthesis? Kahan gayi wo asymmetric synthesis? So then what you planned? Adjacent to this carbonyl carbon, either on this side and either on this side, you placed a chiral environment. Now can you see your substrate in carbonyl group? You placed a chirality where methyl is on above the plane and hydrogen is your below the plane. Now you bring the methyl group. अब आप प्रेफर करके बताइए कि जब आपके सबस्ट्रेट में अपर प्लेन पे मीथाइल ग्रुप है, लोअर प्लेन पे हाइड्रोजन है, जो आपका इनकमिंग ग्रुप है वो किसकी साइड से आना प्रेफर करेगा? From the side of hydrogen, which has a less steric hindrance. So can you see in your product the methyl is attached from the side of the hydrogen? So what you have done in this reaction? If this chiral unit adjacent to your carbonyl group is not present in the substrate, you get 50-50 both the enantiomer. But what you have done? The chiral unit is placed adjacent to the carbonyl group. Now the incoming group will decide to come from the side which has a very lower steric hindrance. So that's mean the formation of pure enantiomeric chiral product is controlled by the presence of chiral unit in the substrate. So that's why it is called substrate control. So if anything is present in the substrate which will help you to lead to the formation of only one type of enantiomer or a product, then that is your substrate controlled first generation reaction. Clear हुआ? That now it will be asked, what do you understand by first generation? तो आपको क्या कहना है कि there must be some chiral unit present in the substrate which will guide the incoming group to attack on the substrate, because the incoming group will only depend upon the presence. Now just imagine this is our seminar room, we have 2 doors. Now if I am inside, I have closed this room. Now I am controlling it, that how anybody can enter to this seminar room, I am controlling it. Okay? But if I am not here, you people are sitting, both the doors are open, anyone is coming, you don't have to say anything. They are entering, they are sitting. But as soon as I arrived here, I don't want that anybody should come here because he or she may disturb me. So I just locked it. Now who is controlling the coming of the students into the seminar room? Right? So in a similar way, if in your substrate no chiral unit is present, carbonyl group is lying in a plane, then the incoming group have a preference to coming from the upper plane or from the lower plane. So that means 50-50% possibility you are going to get racemic mixture. Okay, but if you want to get enantio specific reaction, you must have some chiral unit or chiral auxiliary which is present on the substrate, which will control the formation of the product. So can you see that in reaction scheme we have seen S with G. G is your chiral directing group. The reagent you are using a chiral reagent, methyl magnesium iodide is a chiral. इसमें तो कोई आपकी chiral unit नहीं है. So फर्स्ट जनरेशन तो क्वेश्चन आपको ऑब्जेक्टिव और ऐसे प्रिपेयर हो जाता है कि इफ योर substrate has a chirality, it belongs to what generation? So the first generation is your substrate controlled. Okay, now if you see the chirality is induced intramolecular. आपके सबस्ट्रेट में chiral यूनिट थी, उसने इनकमिंग ग्रुप को गाइड किया और आपके प्रोडक्ट में एक नई chiral यूनिट आ गई. So this is occurring within the molecule. So that means it is intramolecular. The question comes, first generation is intramolecular and intermolecular. आपको इंट्रा और इंटर का डिफरेंस क्लियर है? इंट्रा इज विद इन द मॉलिक्यूल, इंटर इज बिटवीन द टू. ओके, नाउ व्हाट इज द ड्रॉबैक? नाउ यू कैन सी इन होल स्लाइड इन रेड कलर it has been discussed. What is? However, the enantiomerically pure starting materials with appropriate configuration may or may not be available in chiral pool. अब आपके सबस्ट्रेट में जो chiral यूनिट प्रेजेंट है, ऐसा सबस्ट्रेट अगर आपके पास अवेलेबल ही नहीं होगा, will you go with first generation? So first generation की कंडीशन क्या है? That you must have a chiral unit in your substrate. But if your substrate are not available with variety of chiral units, you cannot go for the first generation. So अब मुझे बताइए first generation के relation में कोई query आपको है? Is it understand? After this lecture I am going to ask, because this is a tutorials, you have to participate in it. Okay, so in first generation you know that is a substrate control. Now comes that if my substrate doesn't have any chiral unit, करेगा. Here we are just talking about the getting of one product. Okay, but which one, whether it is from the above, whether it is from the below, this comes with the different auxiliaries. Okay, so it is the blockage. Okay, if we have to answer his question also, I said I have locked this door but I have opened it. I can do the reverse also. If I don't mind that students are going outside from that side, I just close it. I am teaching at the podium, any student can come and he or she can sit. What is the problem? It is only what I have required, what is my desire. So that I am going to explain in evening lecture or morning, where we are talking about phase blocking. Here we are saying out of the two we have to just block one. Okay, but next is which one? It also depend as per your requirement कि आपको जो अब in this molecule, in this product, can you see methyl and methyl are on the opposite side? But if I just want to choose the methyl on the same side, I have to take the substrate as a reverse one. Okay, so whatever the transition state is going to form, that we will have to discuss as per our preference. For that there are different auxiliaries are present. Okay, so if first generation is okay, we are moving to the second generation. Now can you see in second generation what is the substrate? Palvi, क्या substrate है second में? cyclohexanone, ओके, नाउ इन दिस सबस्ट्रेट आपको कोई कायरल यूनिट दिख रही है? We don't have any chiral unit. Then who is going to control the attack of the incoming group? Okay, so what we are doing, अगर तो chiral pool में आपके पास substrate में chiral unit मिल जाए तो best है, अगर नहीं मिला कोई बात नहीं, we move something else. So that something else is auxiliary controlled. So what we do in this, the chiral directing group, the chiral auxiliary is deliberately attached to the achiral substrate which is removable after it has served its purpose. आपने फर्स्ट जनरेशन में क्या देखा कि कार्बोनाइल के एडजेसेंट पे दो ग्रुप प्लेस्ड हैं, एक हाइड्रोजन है और एक मिथाइल है. दे मस्ट बी डिफरेंशिएटिंग, तभी आप उनका यूज कर पाओगे. तो उन ग्रुप का काम क्या था? क्या उन्होंने केमिकल रिएक्शन में कोई पार्टिसिपेट किया? नहीं. उन्होंने तो फेस को, प्लेन को डिफरेंशिएट किया. कैसे डिफरेंशिएट किया? एक साइड मिथाइल है, दूसरी साइड हाइड्रोजन है. ओके, सो दोनों ग्रुप डिफरेंट है तो दोनों की स्टेरिक इन्वायरमेंट डिफरेंट है. सो आपने दो प्लेन को डिफरेंशिएट किया फॉर द इनकमिंग ग्रुप. सो अगर हमारे पास सबस्ट्रेट में कोई ऐसे ग्रुप नहीं है तो हम अपने सबस्ट्रेट में ऐसा ग्रुप प्लेस कर सकते हैं टेम्परेरीली, ताकि वो दोनों प्लेन्स को डिफरेंशिएट कर सके. When it differentiated the both planes, you perform the reaction, and once the reaction is over, you remove that auxiliary, you will get your final product. So if you see the scheme, that if your substrate, you place the A star, that is the auxiliary. Once you place the auxiliary, then you bring your reagent, a chiral reagent, then that reagent is going to add with respect to the auxiliary which is present. Once it is, your product is formed, remove the auxiliary. Now can you see in your reaction, that is cyclohexanone, you react with it phenylalanine, get the chiral auxiliary product. Now what is this? What is this big structure? In a very simple way, कार्बोनाइल को अगर आप अमीन से रिएक्ट करें तो क्या बनेगा? इफ आई हैव अ एल्डिहाइड और इफ आई हैव अ अमीन, व्हाट विल यू गेट? आई गेस आप in your laboratory you get many papers that you are preparing Schiff's bases. You are treating aldehyde and amine, getting carbon nitrogen double bond. So that's mean they are complementary group. Okay, so you find a substrate and the appropriate complementary group, chiral complementary group, and make the auxiliary on the substrate. So starting हमने क्या लिया? That is cyclohexanone. उसमें आपने फिनाइल एलिनोल लिया, यू गेट कार्बन नाइट्रोजन डबल बॉन्ड. नाउ द क्वेश्चन इज कम, हाउ दिस ऑक्सिलरी हैज टू बिहेव. इट शुड बी अ सिंपल वन और इट शुड बी अ वेरी क्राउडेड वन. मनरीत, क्या होना चाहिए? इट शुड बी क्राउडेड वन. अगर मैं आपको कहूं दैट define the whole asymmetric synthesis in one word, तो आपने क्या कहना है? नॉन इक्वलेंस. अच्छा, मैं आपको बड़ा सिंपल एग्जांपल देता हूं. इफ यू हैव टू रिकॉर्ड द NMR स्पेक्ट्रा ऑफ इथाइल अल्कोहल, हाउ मेनी टाइप्स ऑफ पीक्स यू आर गोइंग टू गेट इन स्पेक्ट्रा? इथाइल अल्कोहल में NMR स्पेक्ट्रा में कितनी पीक्स आपको मिलते हैं? बट आई एम सेइंग उसमें तो हाइड्रोजन ही है. व्हाई यू आर गेटिंग 3 पीक्स? इफ द एटम इज सिमिलर, CH3 CH2 OH, but why you are getting 3 different peaks? because you are saying that all the three hydrogen have a different environment with respect to each other. So if with the same atom you are going to create a different environment, you are going to get a different peaks. That's mean you are going to get a differentiation. So similar is the case with asymmetric synthesis, because if you are talking about symmetry, it is 50-50, there is no issue, you are getting all the enantiomer in equal ratio. But if you want to create a asymmetry, that's mean you must differentiate the plane which is above and plane which is below. So that differentiation, that non-equivalence should be there. So how you create a non-equivalence? You are putting a group, a bigger group. So if a bigger group is either present above the plane or below the plane, you don't have to figure out that in final product why this configuration. Just remember here that you are distinguish the two planes for the incoming group. Okay, so what you are doing? In substrate you are going to add one group in such a way that it can create a huge crowding towards one plane, so that the incoming group feels discomfort. Okay, it feels such a discomfort that it try to prefer to come from the other side. Okay, if I am standing here, anybody is coming late after 5 and 10 minutes, I am scolding him or her, but in that side nothing is going to happen, nobody is there, you can come easily and sit from where you want to come. You want to come from this side? जहाँ पे मैं खड़ा हूँ आपसे पूछ रहा हूँ लेट क्यों आए, ऐसे क्यों आए, मैं तुम्हें अंदर नहीं आने दूंगा, so many things. But in that side you don't have any issue, door is open, you are coming at 9:15, come and sit and enjoy the topic. What is your preference? You want to go from that side. Similarly, if in the plane you are creating such a crowdness, bulkiness of the substituent on one side, why the incoming group try to enter from that side? It try to enter from the opposite side where there is no steric crowding or any issue. So if it is preferably coming from the other side, your objective is reached, that you block one face, open the other face, your enantio selectivity and enantio specificity already increased. Okay, so in this case you place the auxiliary, आपने सेकेंड स्टेप में ऑक्सिलरी प्लेस कर दी, उसके बाद आपने रिएक्शन शुरू किया. व्हाट इज द रिएक्शन इन दिस? आपने क्या ऐड किया हुआ? बेस. इफ यू सी, आई जस्ट मार्क दिस इज आर स्टार. दिस आर स्टार और ए स्टार इज ब्लॉकिंग एनी फेस. डोंट गो वाई अबव वाई बिलो, जस्ट सी इट इज ब्लॉकिंग वन फेस. नाउ व्हाट इज द नेक्स्ट स्टेप? यू आर ब्रिंगिंग द बेस. What a base will do? It will abstract the proton. This base is abstracting the proton. You will get a negative charge here which is delocalized in this system. Now you treat your reagent. Okay, that is going to come. Now the reagent is coming from which side? Where you don't have this phenyl HCH2OH group. It is coming from the side which is away from the steric bulk. So you are getting the target product. So that is your pair of diastereomers. Now can you see, in the presence of auxiliary you have a two stereogenic center. रेजोल्यूशन स्टेप यहां पे अप्लाई हो सकता है. आपने एक इनकमिंग ग्रुप के आने से कायरलिटी इंट्रोड्यूस कर दी, एक कायरलिटी आपने एज अ ऑक्सिलरी प्लेस की हुई है, दो हो गए तो क्या हो गया आपका? डायास्टीरियोमर्स. If diastereomers are the product, separate them and then remove your auxiliary, you get your final enantiomeric product. Now can you tell me, it should be intramolecular or intermolecular? Intramolecular. Okay, the chirality is induced intramolecular, क्योंकि आपने सबस्ट्रेट में पहले से ही ऑक्सिलरी प्लेस की हुई है. That auxiliary is now controlling the incoming group. So that is your intramolecular, that is within the molecule. The advantage is what? That in this case you are getting diastereomers which can be separable, okay, but what is the limitation? That the appropriate auxiliary with complementary functional group may not be available. अब आपने तो सबस्ट्रेट चूज किया था कार्बोनाइल ग्रुप, इसके साथ ऑक्सिलरी को रिएक्ट करने के लिए आपके पास कायरल अमीन अवेलेबल थी, तो आपकी ऑक्सिलरी अटैच होगी. सो दैट इज योर कॉम्प्लीमेंट्री ग्रुप. अब उस ऑक्सिलरी में बल्कि ग्रुप भी प्रेजेंट है जो एक फेज को ब्लॉक कर सकते हैं, बट दैट मे नॉट बी प्रेजेंट. सो दैट इज द लिमिटेशन ऑफ दिस जनरेशन इफ दिस सबस्ट्रेट controlled cannot be available. Okay, so can you explain me second last student? हाँ जी, आपसे बात कर रहा हूँ. कैन यू, नहीं आप बैठिए, आगे आगे. अरे आप इस साइड में. हाँ जी, आपसे ही बात कर रहा हूँ मैं. हाँ जी, हाँ जी, क्या नाम है आपका? Arun, can you explain me what is the limitation of this second generation? अब दो ही ऑप्शंस हैं, या आपको नहीं पता है या आप एक्सप्रेस नहीं कर सकते. आगे आ जाओ, मुझे बिल्कुल सुनाई नहीं दे रहा. हाँ, नहीं, इसमें हमें क्या प्रॉब्लम आ सकती है? नहीं, वो बात तो इसकी लिमिटेशन की नहीं है ना, वो तो रिएक्शन की बात आ गई ना. आप ऑडियंस को क्या एक्सप्लेन कर सकते हैं जो हमने लिखा है, व्हाई वी हैव टू गो टू थर्ड जनरेशन? सेकेंड जनरेशन की क्या लिमिटेशन है कि हमें थर्ड जनरेशन पे जाना पड़ेगा? वो तो इसकी एक फीचर हो गया, वो लिमिटेशन नहीं है. लिमिटेशन तो लास्ट स्टेप में है. अच्छा, मुझे ये रिएक्शन देख के बता सकते हैं कि इस स्कीम में हम कर क्या रहे हैं? बैठ के बताओ, बैठो. See, somehow after this program you have to appear for any professional degree. Okay, you have to go for any interview panels and there. वहाँ पे आपको एक्सप्रेसिव होना है. It is not that you are not able to get the question. They can say, okay, this is the screen, this is the reaction. आप इसको देख के क्या एक्सप्लेन कर सकते हैं? See, now I am explaining again, just try to listen. In this case your substrate has no chiral unit, so you are deliberating putting a chiral unit in on it. Okay, for putting a chiral unit on it, that means your substrate and chiral auxiliary must react with each other, then you are going to place it. So I am saying in your substrate you have a aldehyde group, carbonyl group, and the auxiliary which you are putting, you have a NH2. So they are complementary, you are going to get Schiff base. But if that complementary functional groups are not present, how your substrate is going to react with the auxiliary? So this is the first limitation. Okay. The next is, if you are attaching a carbonyl group with any auxiliary, that auxiliary must have some groups which will block one phase, because ultimately if you remember the NMR spectra of proton of ethanol, you said non-equivalence. What is non-equivalence? In center you have either above the plane or below the plane. So if you are not able to create the non-equivalence, then the possibility is 50-50%. So we have to create non-equivalence. Okay. So that's why the appropriate auxiliary with complementary functional group may not be available. So this is the limitation. So we have to move to third generation. So till now first generation is your substrate controlled, यानी substrate control करेगा आपकी asymmetric synthesis को. Second generation is your auxiliary control, जो auxiliary आप introduce करोगे substrate पे वो controlled करेगी asymmetric synthesis को. Now the third one is your reagent controlled. Okay. That's why I said it is not necessary that what you are seeing in this picture, H and methyl, they are going to same or other side. There are some other factors which are going to end, that we are going to discuss. I am just hinting one thing. For example, why I am taking the relevant examples which can be easily understandable to you. If someone is roaming around the seminar room to get his certificate attested by me, I don't know him or her in any respect, okay, but he or she is moving outside to get the certificate attested, I am refusing it because I don't have any connection with them, okay. So I can't trust whether I am doing right or wrong. So this is the one theory which we will follow. But that people or the girl and a boy who is moving around, they have some connection with you. Whether your brother, sister, friend, known to, you came to me, sir, this is my friend, she or he is applying for this admission form, examination form, just attest it. So I will check his document and I will attest. Then how this theory changed? Because you came in between. I have a connection with you, I have a faith with you, I have a trust with you. Then you are emphasizing that candidate with respect to your, because if it is your brother and sister, then how can I say the document will be fake? Okay. So if that connection change my first aspect, so sometimes what happen? The question came, how we have to check whether it is coming from the up phase, whether it is coming from the lower phase. It is not directly what you see in the picture. It is the mechanism aspect. How that mechanism aspect, sometime we are crowding the one plane, second plane is open, then you can say okay, we can go from above side because नीचे जो side है वो काफी bulky है. But if I place a group in a bulky side, that group is more attractive to the incoming group that will go from that side. Okay. Now if I instruct the candidate to come into room by this door, he or she is to come from this door. Maybe here 7-8 students are standing, okay, maybe a table is here, 5-6 chairs are there, that door is open. So it is more comfortable for student to come from that side. Here is so much of blockage, but I am standing here. I tell Tanya, come from this side. She will make a room, or I will make a room that she will enter from this side. But on a general terms, you see that is the open face. Here is the blockage. We should come from that side. Okay, so there are a different mechanism aspect with respect to particular reaction. So here I am just emphasizing first, second, third, fourth generation, what is the main keyword, but exactly how this plane selection is going to occur, it depend upon the functionality which are attached, whether they are directly refusing it or whether they are also helping it to attach the incoming group. Okay, so that will come in the later part. Okay, so that is our third generation. Now third generation is your reagent control. Can you see in your substrate any chirality? आपके substrate में क्या कोई chirality है? No, only double bond and CH3 is present. Okay, so the achiral substrate is directly converted to chiral product by the use of chiral reagent. अब आप इसमें जो reagent ऐड कर रहे हो वो chiral in nature है. तो अब बताओ ये इंट्रामोलिक्यूलर होगा कि इंटरमोलिक्यूलर होगा? इंटरमोलिक्यूलर. द chirality is induced intermolecularly. Now how you compare this generation with respect to for second generation? In second generation you have to add first auxiliary, then after the work done is over you have to remove the auxiliary. So जो दो additional steps है वो आपके इसमें क्या हो गए? Cut down हो गए, because you are directly taking a reagent which is chiral in nature. Okay, now if you see in this reaction, what is this reaction? Have you done hydroboration around the carbon carbon double bond? See, mostly in asymmetric synthesis we are doing the reaction across double bond, because carbon carbon double bond or carbon oxygen double bond both are in plane, both are sp2. So all the reaction which are occurring around these two functional group, they all can be used in the asymmetric synthesis. एल्कीन्स के कौन कौन से रिएक्शन पॉसिबल हो सकते हैं? आप सेंटर में जो बैठे आराम से यही बैठते हैं. कार्बन कार्बन डबल बॉन्ड के अक्रॉस हम कौन से रिएक्शन प्लान कर सकते हैं? ओके, आपने पढ़ा हुआ है मार्कोनिक रूल, एंटी मार्कोनिक रूल. कार्बन कार्बन डबल बॉन्ड इज अ पोलर बॉन्ड. You are always able to get partial positive, partial negative. So कहीं पर भी अगर आप partial positive and partial negative create कर सकते हो, you can do all type of addition reactions there. Okay, so in this case you can add the groups. So hydroboration में आप क्या करते हो? सिंपल आपने पढ़ा होगा ना, हाइड्रोबोरेशन ऑक्सीडेशन. तो आप BH3 लेते हो, दो कार्बन है आपके पास, एक साइड हाइड्रोजन अटैच कर देते हो, दूसरे साइड BH2 अटैच कर देते हो. नेक्स्ट स्टेप में आप उस BH2 की ऑक्सीडेशन कर देते हो तो आपका OH ग्रुप आ जाता है. सो दिस इज अ सिंपल रिएक्शन अराउंड कार्बन कार्बन डबल बॉन्ड. तो अगर इसी रिएक्शन का आपने एसिमेट्रिक वर्जन देखना है तो आपकी एल्कीन प्लेन में है, आप जो बोरोन रिएजेंट लेकर आ रहे हो वो कायरल ले आओ. क्यों? क्योंकि आपको बोरोन में BH3 से सबस्ट्रेट में क्या ऐड करना है? वन हाइड्रोजन. इफ यू आर टेकिंग वन मोल ऑफ substrate, in that case what you are going to add? 1 हाइड्रोजन एंड देन BH2। तो जो BH2 है, जो ये 2 हाइड्रोजन है, इसकी जगह आपके पास कोई कायरल यूनिट लगी हो, so that is going to help in addition। okay, so in this case ये कायरल बोरेन जो है, ये कायरल पूल में एक मॉलिक्यूल है अल्फा पाइनिन। ओके, ये बोथ एनेंटियोमर में अवेलेबल है, पॉजिटिव में भी और नेगेटिव में भी। तो इसको आप BH3 से रिएक्ट करिए, 1 हाइड्रोजन रिप्लेस हो गया आपका, तो आपके पास क्या बन गया, कायरल रिएजेंट। So you are going to get a one particular type of product in this case। इसमें आपकी जो addition हो रही है, can you see what is the addition? Hydrogen and OH from the same phase। अब आप प्रोडक्ट में देखिए, मिथाइल वाली साइड हाइड्रोजन है और नेक्स्ट कार्बन में क्या है आपका OH। So they are attacking on the same phase। अब आप कायरल रिएजेंट भूल जाइए, कार्बन कार्बन डबल बॉन्ड के अक्रॉस सिंपल बोरेन रिएजेंट को ऐड कीजिए, यू हैव अ पॉसिबिलिटी ऑफ 50-50। क्लियर? सो इन दिस थर्ड जनरेशन you are choosing a reagent which is chiral in nature, that's why it is called a reagent controlled reaction। okay, so I again repeat, first generation substrate controlled, you have to remember only keywords, वहीं से आप सारी method बता दोगे। first is substrate, second is auxiliary, third one is reagent। now we move to the last generation, that is the catalyst controlled। now for this generation 2021 the scientists got the Nobel Prize for discovering a new asymmetric organocatalysis। now what is the reaction? the achiral substrate is directly converted into chiral product using achiral reagent but controlled by a chiral catalyst। अब ना तो आपके पास सबस्ट्रेट कायरल है, ना आपके पास ऑक्सिलरी है, ना ही आपके पास कोई रिएजेंट है जो कायरल है, तो अब आपके पास कायरल क्या है, कैटालिस्ट। अब जो कैटालिस्ट आप डाल रहे हो, उस कैटालिस्ट के ऊपर जो भी स्टीरियोकेमिस्ट्री अवेलेबल है वो अब हेल्प करेगी आपको पर्टिकुलर इनेंशियोमेरिक प्रोडक्ट देने के लिए। okay, so now in this case also chirality is induced intra or inter, so always you have to remember, when it is substrate, when it is auxiliary, the chirality is introduced intramolecular, when you are using reagent and catalyst it is intermolecular, because you are reacting them with your substrate। okay, now unlike first, second and third generation, the chiral compound or chiral catalyst is required in catalytic amount only, not in a stoichiometry amount। ये जो chiral compounds होते हैं, आपने HPLC सुना हुआ है, GC, HPLC, chiral chromatography, इसमें बहुत कॉस्ट होती है, इसमें आपको chiral compounds बनाना, परचेज करना, तो कि काफ़ी अच्छा खासा फंड आपको इसमें यूटिलाइज करना पड़ता है। तो अगर आपने सबस्ट्रेट यूज करना है तो वो तो स्टाइकोमेट्री अमाउंट में यूज होगा। ओके, आपने ऑक्सिलरी यूज करनी है, ऐसा तो नहीं कर सकते कि अगर एल्डिहाइड 1 मोल ले लिया और जो अमीन है वो 0.5 मोल ले ली और आपने रिएक्ट कर दी और आपका प्रोडक्ट बन जाएगा। दे शुड बी into 1 ratio, then you are going to get your corresponding shift base। so यहाँ पे आपकी कॉस्ट बहुत यूज होगी, आपकी फंड बहुत यूज होंगे, बट इन दिस केस what required? chiral catalyst, and catalyst does not consume in the reaction, it is only used for lowering the activation energy or enhance the speed of the reaction, providing the space surface where your substrate and reagent can come bind in appropriate and proper orientation, and then the reaction can be proceed. Okay. So if you see the reaction, the catalyst they are using, that is syncodeine. Can you see in bracket what is the stoichiometry? 0.01 equivalent. Okay. Now in this catalyst, what is the reaction? In reaction, you are going to add a group around the कार्बन कार्बन डबल बॉन्ड. आप रिएक्शन देखिए, इस रिएक्शन में आपको क्या शो हो रहा है. अगर मैं कहूं सिंपल अल्कोहल को आपने कार्बन कार्बन डबल बॉन्ड के अक्रॉस ऐड करना है. ओके, अल्कोहल भी छोड़ दो, वाटर ले लो. इफ यू हैव टू ऐड अ वाटर across the carbon carbon double bond, how you are going to add? One side hydrogen, other side OH. Now instead of OH I have a thiol or SH. Okay, so in this product, if I am talking like at this position, one hydrogen and here SR group, they are going to add across the carbon carbon double bond. So how they are going to add? With respect to the chiral unit present in your catalyst. Catalyst has a chiral unit. One side it is hydrogen and one side is either OH. Other bulky group are guiding the incoming group. So you are getting your single enantiomeric product or enantiospecific product. Now what is the important part, why this can get a Nobel Prize in fourth generation? Because you are reducing the cost of the reaction, plus whatever the chiral product you are making, that can be reused because it is a catalyst. It can be recovered or reused. Okay, so basis we will do in the next class. Now if I am now summarizing this, that you have to plan a asymmetric synthesis for any industry, you have to see what is the availability with you. If you have a substrate, then you can move with the first generation. If you have a substrate but that don't have any chiral unit and you have some complementary group available with you, then it is the second. Okay, but if you have any chiral reagent is present, then it is the third one, but if catalyst is present, it is the fourth one. So if now you have to label number one, now in whole scenario, if you have a keywords with you, you can explain full theoretical concept. What is full theoretical concept? What is your objective? Out of the two enantiomer, I want to get one in majority. In every case, what is your objective? Either two are available, four are available, you want to get one enantiomer in majority, that is your objective. So how this objective can be achieved? If generation 1 by substrate, generation 2 by auxiliary, generation 3 by reagent, generation 4 by catalyst. Okay, this is the only concept where now can you tell me, if we go by any chemical reaction, what we required? Substrate, reagent, catalyst, then you are going to get your any product. तो सबस्ट्रेट कभी भी आप 1 2 3 स्टार्ट करके देखिए, सबस्ट्रेट 1 जनरेशन, ऑक्सिलरी 2 जनरेशन, रिएजेंट 3rd जनरेशन, कैटलिस्ट 4th generation. You are not going to miss any objective question if you are going to get this way. Clear? If you have any query regarding this, you can ask. क्लियर हो गया सबको? ऐसा तो नहीं है कि सब ऊपर से चलेगी, यानी इस 1 आवर के मेरे लेक्चर का कोई बेनिफिट नहीं होगा। आई डोंट वांट दैट यू रिमेंबर एनी स्कीम्स रिएक्शन, बट कहीं भी आप एज अ केमिस्ट, केमिस्ट्री डिपार्टमेंट से आप कहीं भी हो, आपसे कोई जर्नल कहे भी आप लोग क्या करते हो, यू विल से वी आर प्रिपेयरिंग मॉलिक्यूल्स, वी आर प्रिपेयरिंग डिफरेंट मटेरियल्स। सो वो उसमें कहीं भी आपके तो एक रिएक्शन में पॉसिबिलिटी हो जाती है इतने एनहेंसोमर की, तो क्या करोगे आप? मुझे तो सिलेक्टिव मॉलिक्यूल चाहिए, क्योंकि नीड आपको बताई है। अगर एक प्योरिटी में नहीं दोगे तो कोई उसको रेसमिक मिक्सचर को तो कोई एक्सेप्ट नहीं करेगा। ओके, ठीक है।
We have a 5 minutes short break. We now move on to our next lecture by our distinguished international expert, Professor Sanjay V. Malhotra from Oregon Health and Science University. He will be presenting on molecular imaging in drug development with a focus on introduction to multimodality molecular imaging. This session will take us into the exciting world where chemistry meets imaging and innovation.
Thank you, Tanya. And my apology, I will have to change a little bit again. In order to do this, I will go over on pharmacology and toxicology, and then I will bring to imaging. So, but imaging is coming. Stay tuned. All right, good morning. आप लोगों को आराम तो मिल रहा है ना कि मेरे लेक्चर सुनने के बाद में इतना बोरिंग हो जाता है या इतना ऊपर से भारी हो जाता है. ठीक है. No, so what as I was saying कि मैं so you saw we learned about the कहाँ से शुरू हुआ, किस तरह से शुरू हुआ, जो chemical looking from the lens of chemistry towards the biology. एक बायोलॉजी को देखना है तो केमिस्ट्री के लेंस से, कि केमिकल्स के लेंस से उसे देखने की कोशिश करें. So we saw chemical solutions for biological. We looked at the biological space, but the focus is experimental therapeutics, drugs. So I showed you कि, can you hear me? Yeah. When you look at this, is just showing you a few-year-old numbers. Okay, if you have Yeah, no, this works. मैं चलते हुए बात कर रहा था तो okay, it's working now. So as I was telling, and as you saw today, as to develop a drug, to discover a drug, it's a multidisciplinary enterprise. Chemistry, biology के अंदर भी आप देखें pharmacology, toxicology, disease biology, understanding what is immunology, understanding how assays are developed, understanding how models are developed, if there are models are right or wrong, in vitro models, cell line models, animal models, and even within the animal, what type of animal models you want to create, and then PDX, patient-derived xenograft models, ex vivo human models, taking the human samples and creating a model. Each one of these is an expertise. Is a discipline. उस सब के बावजूद you have to juggle all of these things to make sure that the drug that you design, the things that you design, has the effect that you are looking for without causing any toxicity or damaging. ऐसा नहीं है कि एक चीज़ से तो ठीक कर दे और दूसरे से मार दे. So इन सारी चीज़ों को, and I'll show you how multiple you are juggling multiple of these things together. So and therefore, in order to bring one good drug to the market, this chart, as you start with, for a potential new medicine, just the basic research. You find out discovery. You do preclinical, all the things that I'm telling you, all the preclinical part, and I'll just show you briefly about the clinical side. When you do, then you start phase 1, phase 2, phase 3 clinical trials, and you do not your drug may fail because of it has a property which is not suitable for human consumption, and it may just drop out. So very lengthy, highly resource intensive, multidisciplinary and long process. So if you think, for example, if there are several thousand candidates, molecules which are being tested, maybe only 4 or 5 will enter the clinical trial, and maybe 1 will enter into the commercial world, which will be approved by that. So it's a very, and why is that it takes long time? So just look at here, for example. So drugs could fail for multiple reasons, and I have tried to give here, for example, so this is the, this is maybe a little bit still an old data, but look at this. 39% of the drugs fail because their PK, their pharmacokinetics properties are not good. So when they get to the point of the clinical trial, they fail, or something happens, they fail. 30% of the time, there could be lack of efficacy. What you see, the drug is effective. Drug is effective in your Petri dish model, in vitro model. You can take into animal model, it may be effective, but when it comes to human, you don't see the same effect, whatever be the reason. So 30% of the drug fail because of that, because what you see, the properties which are working in ex vivo human models, they don't pan out in the clinical trials, or so drugs fail. And you see there are various other, the animal toxicity, they are very toxic, they don't go further. And then 5% could be miscellaneous reason, adverse effect, you have many side effects which you are not expecting, or there could be 5% with the commercial reasons, but there could be many reasons. So even though you start with the hope that research part, that drugs are working, but when it gets to the humans, the results are not the same. Now, among many of these reasons, one of the reasons where these things do not pan out, and as you see here on the left is the distribution, for example. Are they just, so let me take to the next slide. Look here, for example, you take, so I briefly mentioned yesterday, but let's, for example, you take an oral drug. You take a drug, it has to be absorbed in the intestine, right? Because it has to be absorbed, then it has to be distributed throughout the body. And to reach, and somebody asked me yesterday, how does it reach to the place where it's supposed to be, right? So it has to, the process, the mechanism, the route of delivery should be such that it reaches to the intended place that you are interested in. So and see, this is basically what happens. So you take a drug, it goes to stomach, as I mentioned, oral, in the case of oral, it goes to the stomach, because of its acidity, it breaks down, and then it gets into the plasma and it gets distributed. And in there, as I showed yesterday, the pH is variation, a lot of variation of pH, and if you have a small molecule, its property has to match with that. It has to be soluble, it has to be bioavailable, biologically available to the site of interest. If it is not available, you will not see the drug effect, because वहाँ तक वो पहुँचा ही नहीं है जो अपने action कर सके. So what happened? It enters so liver enzymes, they metabolize, they break it down to, because those enzymes break it down, the food that we eat, in this case, for example, the drug, and then elimination. Some metabolites which will go this way into the distribution, into the plasma protein, into the bloodstream, and then it gets distributed throughout. This is the distribution process. So this is a very simplified way. Now, this is the oral route. IV, many drugs are not suitable to be taken orally because their properties are such they cannot be, I mean, if you take, they will break it down, so जो उसका active ingredient है वो intact नहीं रहता है. Then either they are given to the bloodstream or given to IV. So through the IV, this route, for example, IV में जब देते हैं, it enters into the plasma, blood, and it gets distributed very quickly. And IV के अंदर जब देना होता है, so see key points I'm telling you, and I'll connect in my subsequent slides. Goal is कि drug has to reach to the site of interest. अगर वहाँ पहुँचा नहीं तो उसका असर नहीं होगा. Now, if you give the oral route, it gets metabolite, it breaks down, goes to the various part of the body. Various parts, they have different functions because there are hundreds and thousands of enzymes जो catalytic reaction कर रहे हैं उस molecule के ऊपर, and it gets distributed or it gets cleared, elimination process gets cleared through whichever route. IV, so oral route के अंदर usually the concentration of the active drug is higher. IV is highly effective, IV के अंदर डोज कम हो तो भी क्योंकि वो काफ़ी है to reach the sites because it gets into the plasma, gets into the blood and blood circulates throughout the body, it gets circulated पूरी body के अंदर. So therefore the reason I'm mentioning is if you next time if you see कि oral pill का size है इतना है, उसमें पढ़ो कि active ingredient कितने हैं, right? और दूसरा when you see the same match on IV के अंदर जो दिया जाता है कितना है, तो usually the concentration will be smaller because small concentrations is enough to reach the site of action. Oral में लेके उसका काफ़ी हिस्सा तो eliminate हो जाता है because of the enzymatic reaction that happened there. So understanding how all of these things, so how the drug has to be absorbed, it has to be distributed, it has to be metabolite, break down to metabolites, pieces that reaches to the site of where action where it needs to be, and then जब हो गया उसका function जो उसके metabolite आगे बने, they are eliminated. So the process absorption, distribution, metabolism and excretion. ADME is an extremely important subject, and for chemists it's relatively easier to understand compared to biology. Because what you are doing is basically the physiochemical properties of the molecule and what mechanistically they will be break down in various parts of the body. वो अगर absorb हो रहे हैं तो उसकी physiochemical property कि they get absorbed. If they are distributed, then they are distributed through some mechanism कि they survive through that mechanism. ऐसा नहीं को टूट गए, they break down, the molecule is broken down. And then metabolites, वो metabolites अगर उनके टूटते हैं तो क्या बनता है उससे? So all of these things and elimination or clearance, they get eliminated. So all of these are relatively easier for a chemist to understand क्योंकि वो it's all around physiochemical properties. You design your drug in such a way, you look at the physiochemical properties of the drug, you can manipulate, you can play around with this thing. So this is what quote unquote is the field of pharmacology. So pharmacology basically is the science of how drugs and biologics act on biological system and how the system responds to it. Okay. And clinical pharmacology basically is when it's the clinic is basically applies to the basic science and principles of pharmacology together. So when you look at pharmacology, it is your basic scientist is looking at from the concept of trying to understand what the drug will do, how it will do, and try to manipulate the drug in order to discover good thing, and then translating that information to clinical side. So think for a second, आपने ये सुना होगा सेम कि कई चीज़ें जो हैं ओरल से दी जा रही हैं, कई जो हैं डर्म से दी जा रही हैं, राइट, स्किन लोशन जो दी जा रहा है, कई चीज़ें जो हैं आईवी से दी जा रही हैं, इंजेक्शन से दी जा रही हैं मसल्स के अंदर, राइट. आस्क योरसेल्फ व्हाई सबको ओरल क्यों नहीं दे देते या सबको आईवी क्यों नहीं दे देते, राइट? सिंपल, व्हाई? It's based on the properties. Based on the properties of what you're, and multiple things. One is the property and one how quickly and it reaches. Now, I will come to that, and I was going to mention a point, but I will come back to that. So, basically, experimental pharmacology, it's preclinical study. Can you take care of understand how, again, ADME talks, absorption, distribution, metabolism, and excretion, how it happens in various of these models. And clinical pharmacology, inside the clinic, किस तरह से किया जाता है कि how the drug will behave, how whatever the candidates will behave. So clinical study are done. So that's why when you hear phase 1, phase 2, phase 3 की जाती है study, I will come to that, what the difference between those phases of studies are. But clinical studies are done in order to first make sure that it is performing as intended before FDA or any regulatory agencies approves and वो masses को easily commercially available हो. So that part of the study is basically what is. So again, I'm giving you this thing again and again just to hammer the definition and understanding of it. So pharmacology again is the science of drug and medication. Specifically, it is the interaction of, so I will come to that, how body reacts, what body does to the drug or medicine, and the second thing is what drug does to the body. So pharmacokinetics and pharmacodynamics, two different terms. आपने ड्रग लिया, ड्रग ने जाके बॉडी के अंदर क्या किया, और आपकी बॉडी के अंदर जो एंजाइम्स या बॉडी के अंदर उसने ड्रग को क्या किया, दोनों के अंदर. सो that's the difference between the two. So if you look at, there are multiple steps. Again, specific, again, the rate that a chemical is absorbed or distributed, understanding of that, that's again pharmacokinetic. The rate and pathway of drug metabolism. अगर वो जाते हैं लिवर में, किस स्पीड से जाते हैं और क्या मेटाबोलाइट्स बनते हैं, किस रेट से बनते हैं, किस रीत से वो डिस्ट्रीब्यूट होते हैं, अंडरस्टैंडिंग ऑफ दैट. एंड अगेन द प्लाज्मा कॉन्सेंट्रेशन ऑफ ड्रग over the time. And this is again, when I say over the time, can anybody think of why I'm saying over the time? Drug plasma distribution कितने time में होता है, over the time how it gets distributed, why is that important? So फर्स्ट कीजिए, ड्रग लिया, ले तो लिया, मगर उसकी कॉन्सेंट्रेशन जो है, whether through oral route or through plasma or the IV route. Let me step back. कि ड्रग जो है 5 मिलीग्राम पर किलोग्राम बॉडी वेट चाहिए उसका असर करने के लिए IC50, if the concentration which will kill the 50% of, has the 50%, that's IC50. So if you take the drug, if a person takes the drug and required concentration is, next time आप जब देखें, just flip the, नहीं मैं देखा, मैं दवाई नहीं निकाल रहा, मैं सोचा था कि मेरे पास में दवाई है तो आपको दिखाऊं, कि you will see, I mean, this is again, it will automatically, you are self-educating, because even when I take a food, I like to read and see what the ingredients are, always. ये आदत पड़ गई है. I look at the ingredients, चाहे food के अंदर भी ingredients क्या हैं उसके, कितने % carbohydrate है, कितने % protein है, just as a chemist, I always look at that. ये है कि पराठों पे लिखा नहीं होता, नहीं तो मैं पराठों पे भी पढ़ूँ मैं क्या हुआ है. But point being, that connects you कि इसके ingredient क्या हैं, कितने carbohydrate, protein, that's one part. Anything that you take, even I just go and buy shaving cream, I look at back कि उसके अंदर कौन से menthol कितना है और वो सब क्या है. It just as a chemist, मेरी को आदत पड़ चुकी है. Drugs अगर आप देखें, कोई भी आपको दवाई का पत्ता मिलता है, just turn it around and see उसमें कौन से ingredient कितने % है, right? So let's say if the drug for its intended effect is 5 mg per kg, 5 milligram per kilogram body weight चाहिए उसका, बट उसकी प्रॉपर्टीज ऐसी है कि ड्रग लिया, वो सॉल्युबलाइज नहीं हुआ, और मेजॉरिटी ऑफ ड्रग है वो आपका किसी भी रूट से वो बाहर निकल गया, उसका असर होगा नहीं होगा. 1 पॉइंट. सेकंड, तो उसमें अगर 50-90% निकल गया तो 10% बचा. अब 5 mg पर केक अगर उसका चाहिए और आपने 5 mg पर केक लिया तो उसमें से 90% तो निकल गया बाहर, वो क्लियरेंस हो गया, यूरिन रूट से, जिस रूट से वो बाहर निकल गया. 10% बचा तो उसका इंटेंडेड से होगा नहीं. फिर लेंगे तो बोलेंगे जी दवाई काम नहीं कर रही. इट्स नॉट दैट दवाई काम नहीं कर रही, बट एक्चुअली मार्केट तक आता नहीं है, बट दैट्स कि मार्केट, दैट्स हाउ दीज थिंग्स आर स्टडीड. Second thing for a second, okay, that is one part. Second thing, okay, let me ask you this question. आपने सुना होगा कि डॉक्टर से आपने ली दवाई, ओके जी, कि आप दिन में 2 बार लीजिए या दिन में 3 बार लीजिए या एवरी अदर डे लीजिए. Why? Think, क्यों? वाई एक पिल को 3 बार क्यों लेना पड़ रहा है, या एक ही बार क्यों लेना पड़ता है, ऑल्टरनेट डे पे क्यों लेना पड़ रहा है? राइट, सिंपल क्वेश्चन. आई एम श्योर आप लोगों ने सुना हुआ ये, टेक ट्वाइस अ डे, 3 times a day, or take these many pills or anything. As a chemist, convert that back. Why? So now you see, it will be good to read the ingredient. It will be good to read कि उसके active ingredient क्या है. And so here is the reason. ड्रग की हाफ लाइफ जो होती है ना, क्लियरेंस हाफ लाइफ, कितना जो पहुंचता है. सो इफ उसकी हाफ लाइफ के बेसिस पे डोजेस उसके डिसाइड किए जाते हैं. इफ यू टेक द ड्रग, हाउ लॉन्ग इट विल स्टे इनसाइड द प्लाज्मा, इनसाइड योर बॉडी. 50% ऑफ द, उसका 50% कम होता जाएगा, उसका 50% कम होता जाएगा, राइट. सो मगर इफ ड्रग जो आप टाइलेनॉल, वेल आई डोंट नो, टाइलेनॉल में यहाँ पे है एसिटामिनोफेन, आई डोंट नो कौन से ब्रांड नाम से यहाँ बिकती है, नॉट एनासिन के अंदर तो डिफरेंट होता है, बट यू नो सिर की दवा, सिर दर्द की दवाई फॉर एग्जांपल, राइट. सो उसका जो है हाफ लाइफ जो है प्लाज्मा रहने के बाद में 50% रहेगा. अगर 1 ग्राम लिया, 1 ग्राम इज टू हाई, पहले से 100 मिलीग्राम लिया, उसका अगर 6 घंटे के अंदर 50% उसका क्लियरेंस हो गया, तो बाकी आज 6 घंटे बाद में उतना ही बचेगा, कम बचेगा. उसके 6 घंटे बाद में और भी उसका भी 50% कम हो जाएगा, राइट. तो मगर बॉडी को उसका इफेक्ट करने के लिए आपको पूरा 100 मिलीग्राम ही चाहिए. एंड दैट्स व्हाई डॉक्टर से एवरी 6 आवर्स कि आपकी बॉडी के अंदर जो है उसकी कॉन्सेंट्रेशन मेंटेन रहे, क्योंकि उसका क्लियरेंस जो होता जाता है, बॉडी गेट्स क्लियर. सो दैट इज हाउ जो डोजेस के बेसिस पे डॉक्टर 2 बार लो या 3 बार लो. अगर डॉक्टर के इसको इतनी बार लो, उसमें घबराने की जरूरत नहीं है. नाउ एज ए केमिस्ट, नेक्स्ट टाइम व्हेन यू हियर, उसको आपको बताना चाहिए कि जस्ट टू मेंटेन द कॉन्सेंट्रेशन इनसाइड द बॉडी. डॉक्टर साहब एक ही बार में क्यों नहीं खत्म करते, 3 बार मैंने क्यों लेनी है दवाई? डॉक्टर क्योंकि उसका प्रॉपर्टी ऐसी है कि वो क्लियर होता जाएगा. ड्रग अगर क्लियर होता क्या, बॉडी में नहीं, बॉडी में नहीं रहा तो उसका फर्क असर महसूस नहीं होगा. सो दैट्स हाउ दीज थिंग्स आर डिजाइन. सो ADME talks, now you can see as a chemist is relatively easier to get into that field, understand and be able to master that field, because it's all around physiochemical properties. And as a chemist, if you look at the structures of the molecules, जैसे मैंने स्ट्रक्चर दिखाया था, the periodic table, these are all atoms, उनके कितने इलेक्ट्रॉन्स है, कितनी आयनिक पोटेंशियल किसकी क्या है, वो यू कैन ड्रॉ ऑल ऑफ दिस. सो फिजियोकेमिकल कितने उसके अंदर कौन से फंक्शनल ग्रुप्स हैं, दे आर रिएक्टिव फंक्शनल ग्रुप्स आर नॉट, ऑल ऑफ दिस बिकॉज दे आर इंटरैक्टिंग विद एंजाइम्स. सो एनीवे, लॉन्ग स्टोरी शॉर्ट, दैट्स द बेसिस ऑफ हाउ डोजेस क्या हैं, कितने टाइम लेना है, whether oral route, whether IV route, those are the things that are decided based on ADME talks. So if you look at just the pharmacology areas हैं, ड्रग composition क्या है, उसकी properties क्या है, ड्रग का सोर्स क्या है, इज इट अ सॉलिड और इज ए लिक्विड जो आपको दिया जा रहा है, IV के सोर्स पे या कहीं भी दवाई भी ली जाए. सो ड्रग अगेन, ड्रग डिजाइन, दिस इज अगेन कि उसकी प्रॉपर्टीज क्या है, इंटरैक्शंस, व्हेन द ड्रग गोज इनसाइड, how it is interacting with enzymes, or if there are multiple drug, people are taking multiple drugs, how there is drug-drug interaction. Pharmacokinetic clearance of 50% availability of one drug versus if multiple drugs are, देखें दूसरे का असर कितना होगा. कई बार होता है ना, वो कहते हैं कि डॉक्टर अच्छा ये वाली दवाई भी लो और साथ में ये वाली दवाई भी लो, और मे बी सम टाइम मल्टीपल दवाई दे, तो वो उसको बैलेंस करने की कोशिश करे. सो दैट्स द क्लिनिकल फार्माकोलॉजी. उसको बैलेंस करने की कोशिश करे कि ये दवाई इतना असर करेगी, उसके साथ में आप दूसरी दवाई लेंगे तो उसके ऊपर क्या इफेक्ट करेगी. बट ऑल दीज डॉक्टर के यहाँ पहुँचने से पहले प्री क्लिनिकल बेसिक साइंटिस्ट लाइक योरसेल्फ, दे डू ऑल द डिस्कवरी पार्ट. Doctor only implements. That's why clinical trials happen, to do that. But see, within pharmacology, kitni interaction, chemical biology, as I've been talking, molecular and cellular, similar metabolite banenge, how distribution will happen, excretion will happen. These are all biological mechanisms. Organ systems is working on antipathic capabilities, signal transduction, medical application, therapy. All of these are come under pharmacology. So, as I mentioned earlier, Pharmacology is combination of pharmacokinetics and pharmacodynamics. Pharmacokinetics, as you see, to study how an organism affects the drug, what body does to the drug. क्योंकि बॉडी के अंदर एंजाइम्स हैं, ड्रग जब अंदर आया तो उसने उस बॉडी ने क्या किया, उसके वो तोड़ मोड़ के कैसे मेटाबोलाइट उसका बनाया, उसका उल्टा कि ड्रग ने बॉडी के ऊपर क्या असर किया. दैट्स फार्माकोडायनामिक्स, द स्टडी ऑफ हाउ ड्रग अफेक्ट्स द ऑर्गेनिज्म. एंड नाउ यू कैन अप्रिशिएट कि दोनों में फर्क है, क्योंकि बॉडी के अंदर आपके जो एंजाइम्स है वो उसके ऊपर अटैक करें, नॉट अटैक बट दे विल इंटरैक्ट, दे विल डू केमिस्ट्री देयर, राइट. Drug comes in with its own property inside the body, so drug is trying to do something in there. So उस पार्ट को समझना कि बॉडी के ने ड्रग के ऊपर क्या किया, that's why हाफ लाइफ अगर उसकी है ड्रग की, that's because बॉडी उसको तोड़ रही है या उसको क्लियर कर रही है. So that's pharmacokinetics. What drug does to the body is pharmacodynamics. So the understanding of each one of those is extremely important when you design a drug to see कि किस तरह से उसके डोजिंग रेजिमेंट बनेंगे, क्या प्रोटोकॉल्स बनेंगे. See, okay, I have this method. I didn't realize I already have that. Both together influence the dosing. What benefit will be there? What will be the adverse effect? वो कहते हैं ना कि ये कुछ यहाँ पर साथ में स्ट्रॉन्ग दवाई या साथ में नौजिया भी हो जाता है. सुना होगा आपने ये सारे टर्म जो है, these are common terms. थोड़ा होता है मगर उसके कारण थोड़ी नौजिया होता है, उल्टी भी आ जाती है, बट ड्रग इज इफेक्टिव बिकॉज दैट्स द साइड इफेक्ट, राइट. बट देन यू बैलेंस के कि बेनिफिट वर्सेस द साइड इफेक्ट. द साइड इफेक्ट शुड नॉट बी टेक ओवर द बेनिफिट साइड ऑफ इट. सो दैट बैलेंसिंग इज व्हाट द साइंटिस्ट डज. दैट बैलेंसिंग इज व्हाट ए फार्माकोलॉजिस्ट डज. ट्राई टू फिगर आउट what is the fruitful balance which allows for drug to come out, to be move forward for preclinical से clinical trial से लेके commercial उसके अंदर, so that intended use is humans. अगर humans के लिए drug बन रहा है, intended use is humans, so they can benefit from there. And that's why जो पहले मैंने दिखाई है, that's why it takes a lot of investment. Just imagine, पहले तो स्क्रीन करना, as I was telling you yesterday, you do high throughput screen, you take thousands and thousands of compounds, now millions of compounds, and with in silico you can start with even billions of compounds to design computationally and bring it down to an actually 10,000 or several thousand. I mean, we have in our collection million, 2.25 lakh करीब हमारे पास में हैं compounds हैं that we never needed, we screen those. पर करते करते जब यहाँ तक पहुँचा, उसके बाद में वहाँ से लेके जाने के अंदर इट्स एन एक्सपेंसिव प्रोसेस एंड हाईली साइंटिफिकली रिगर चाहिए होता है, क्योंकि आफ्टर ऑल इट्स फॉर ह्यूमन यूज. इट हैज़ टू बी डन प्रॉपर्ली. यू कैन नॉट मेक ह्यूमन एज ए गिनी पिग कि उनके ऊपर ट्रायल करते हैं, चलो ये काम नहीं किया दूसरे करें, पर जिनके ऊपर किया उनका बेचारों का क्या होगा, राइट. सो दैट्स व्हाई इट्स ए लेंथी प्रोसेस, इट्स एन एक्सपेंसिव प्रोसेस, एंड इट्स ए हाईली interdisciplinary process where many disciplines come together. So this is basically what is a field in itself. ADME talks comes with it, toxicology, and I'll cover a little bit of that too. But this is what is the absorption, the process of a drug entering into the system, circulation from the site of administration. So site of administration means it could be oral, it could be IV drug, it could be dermal. उस साइट से लेने के बाद में वो किस तरह से एब्जॉर्ब होता है, दैट्स एब्जॉर्प्शन पार्ट. सेम वे लुक एट द डिस्ट्रीब्यूशन, द हाउ इट इज disseminated, how it is distributed throughout the body. And then metabolites, how in the liver, for example, how liver enzymes they break it down, क्या metabolites बनते हैं, understanding of that, it's a huge field by itself. Each one of these is a field, is a scientific field. And then excretion, how whatever is left over, how it gets cleared. क्योंकि अगर वो क्लियर नहीं हुआ, बॉडी के अंदर ये accumulate होता हुआ करते तो वो टॉक्स साइड इफेक्ट्स करेगा, टॉक्स करेगा. तो हाउ इट गेट्स ब्रेकन डाउन बाय द एंजाइम्स एंड हाउ इट गेट्स क्लियर, ईच वन ऑफ दीज अंडरस्टैंडिंग is a lengthy process. So I can go over, I'll go over one or two, and then I have slides which you'll get to. So I will, at least you'll get the concept of, yeah. So let's talk about absorption. Now, main routes oral, dermal, IV जो routes हैं उसमें when through any one of these routes, if the drug is taken, how is it absorbed? Comes back to again the physiochemical properties of the drug. What are the physiochemical properties of the drug? So if you look at, just out of curiosity, let's say if you take any class of drugs, you look at the drugs, I mean you can go on FDA site, or there are so many reviews now. Just if you just Google and you put chemical structures of cancer drugs, you'll get a whole chart. Look at, as a chemist, look at those structures. There are lengthy single chain, there are very complex natural products, there are compounds which are many the chiral centers, there are compounds which are ADC, antibody drug conjugate, small molecule and antibody, they are put together. So there are varieties of these. How the drug is administered, orally, dermally, through IV, and once it is done, how it gets absorbed. It has to first absorb to be distributed, right? So उसका absorption process क्या होगा? Just whether it's a passive diffusion or active diffusion, it is through endocytosis. If it is coming through, how is drug? उसको सिर्फ समझना. That's why I mentioned each one of these ADME is a huge field by itself. And the advantage, yesterday and before yesterday, people asked me, how is it that you are a chemist and you are doing all this disease biology, cancer biology, but now you see, because you just follow the properties. You just follow the properties and just a little slow by, try to look from your chemistry lens. Try to look at these things from your chemistry lens. You will see, before you know, एकदम से वो ट्यूबलाइट ऑन होनी शुरू होती है. हाँ, अब धीरे धीरे समझ में आ रहा है सब कुछ. So not to be avoiding, not to be ignorant कि भाई हम तो chemist हैं, हमने इससे क्या लेना देना. No, you have the advantage that the others don't have. You have the advantage that others don't have because you can actually look at from the chemistry lens and you can try to sense make. अब ये जो है मैंने आपको explain किया. A biologist will explain the same thing differently. Okay, वो enzymes break it down. But since most of you are chemists, I'm trying to tell you how to take advantage of your chemistry knowledge. Look at it. All it comes down to is, अगर वो absorb हो रहा है तो कैसे absorb हो रहा है? उसकी physiochemical properties. And you are a chemist. You can better understand than a biologist. उसकी physiochemical properties क्या है? Right? After all that is happening is small molecule versus large molecule. It's all chemistry. Right? So if you look at that, that is itself is a huge advantage as a chemist you have. So next time when you see these things, don't shy away and don't go, bhai, ये क्या पहाड़ आ गया? No, ये पहाड़ नहीं है. You can easily make them a straight line. अगर पहाड़ भी है तो उसको सपाट सड़क बना देंगे आप. It's very easy. But just don't shy away. Don't shy away. एक नहीं तो 2 बार करना पड़ेगा, नहीं तो 4 बार, नहीं तो 10 बार पढ़ना पड़ेगा. But look from the chemistry lens. The lens of chemistry, if you start looking at these things will start to come, and then you'll say, aha, this is really not that difficult. It's really easy. We can really get to understand. ठीक है, पढ़ना पड़ेगा. मैं यहाँ पे खड़ा होकर बोल रहा हूँ because I started many years ago in reading. But all I'm saying is, as a chemist, you have the advantage, which biologists don't have that. They have different advantage, but not the ones that you have as a chemist. So absorption, how it is absorbed, where it's absorbed, how it's absorbed, that plays, as you can see, an important role. Same way distribution, how it gets distributed. If it is getting distributed, as I was telling you yesterday, body, and I think I have a slide I'll bring to that, different parts of the body had different pH. So उसमें कितनी चीजें होंगी? Either your compound is stable, it will survive as is, or it will break down. So you have to understand that if it breaks down, it is not the active ingredient which is needed, which means stop. ये आगे compound काम नहीं करेगा. You need to change the structure of that. You design. That's how the drug discovery is done. Okay. If it passes through that, distribute करने के लिए, as I mentioned, from stomach में 2 pH, as goes up from 3.8 to 7, 8 pH, तो उसकी basicity बढ़ जाएगी. Is it soluble or not soluble? If it is not soluble, it will not go further, right? So solubility is important. and therefore different formulations. Now the reason I'm saying is all these things can now you connect to throughout up until now what you have seen. If you have not asked those questions, कि कुछ चीज़ें हैं वो कहते हैं जी ओरल ले लो, कुछ है जी इसमें मिला के ले लो, राइट? क्यों? सॉल्युबिलिटी, अगर सॉल्युबल ही नहीं होगा वो अंदर जाके बैठा रहेगा, उसका असर नहीं होगा, राइट? सो वेदर इट्स सॉल्युबल और नॉट, यू कैन अगेन केमिस्ट्री, राइट? इफ इज इट बायो अवेलेबल, जो उसका एक्टिव इंग्रेडिएंट विच इज नीडेड टू बी इफेक्टिव, उस फॉर्म में वो अवेलेबल है या नहीं है, अगेन physiochemical property, again chemistry. So that understanding, so this is basically, look at this, why I put this down here. The researcher examines where the chemical travels to. When it's distributed, when it is passing through the travel route, in that route कौन कौन से उसको चीज़ें मिलेंगी, किस तरह के एंजाइम्स मिलेंगे, क्या पीएच होगा, जिस भी रूट से वो जा रहा है क्या एंजाइम्स हैं, क्या पीएच होगा, उसमें वो सर्वाइव करेगा या नहीं करेगा, मॉलिक्यूल वो ब्रेक डाउन तो नहीं कर जाएगा, वो कितनी जल्दी से क्लियर होकर दूसरे रास्ते से नहीं चला जाएगा. सो यू सिंपली फॉलो दैट. सो जस्ट अंडरस्टैंडिंग ऑफ दैट, दैट्स व्हाट द रिसर्चर डज, कि वो केमिकल सर्वाइव करेगा उस कंडीशन में. दैट्स अगेन ओनली द डिस्ट्रीब्यूशन पार्ट. सेम वे, सेम थिंग, नाउ यू अंडरस्टैंड व्हाट, सेम वे मेटाबोलाइट बनेंगे या नहीं बनेंगे, that depends on के which route if it is taking through. उसके अंदर like intestine has different enzymes compared to liver, plasma, different enzymes. उन enzymes का उसके ऊपर क्या असर होगा, again what body does to the drug, pharmacokinetics, right? So उन enzymes का आपके मॉलिक्यूल के ऊपर, ड्रग के ऊपर क्या असर होगा, उसकी जो स्टडी है उसे समझना. सो मेटाबोलाइट बनेंगे या नहीं बनेंगे, तो कि बनेंगे तो कौन से बनेंगे. इफ इट ब्रेक्स डाउन टू मेटाबोलाइट विच इज आल्सो एक्टिव, परफेक्टली फाइन. इफ इट्स नॉट, इफ इट कन्वर्ट्स इनटू समथिंग विच इज नॉट एक्टिव, then okay, this is not the right drug, you change the structure. So that's why it takes 10 to 15 years to bring a good drug to the market. And then when all of this is done, how जो भी उसके side products बनते हैं, how they get clear from the body, the excretion part. So those things, now you can understand why all of these, the importance part, why this is important. Any question? Is what I'm talking making sense? Don't hesitate. Any question? Anything else you want to know? So many drugs, I gave you the simple properties, क्या होता है, कैसे होता है, क्यों होता है, just based on the pharmacokinetics, just based on the ADME properties of the drugs. So now you can appreciate why ADME is an extremely important thing, because if you don't know those properties, a good drug cannot be designed, and even if a drug is designed, it will not be effective. And again, now many cases, now it's a complex machinery. When the drugs are designed, let's say all of these things are good, but in some cases, in many cases that you hear that the drug still doesn't work in certain patients, or there is a side effect. So let me explain to you why that happens. So let's say if there is a drug for a person who has no other morbidity, no other complication, तो जैसा बताया theoretically it should work that way. Now imagine the same drug is given to, and again how these things are effective, and usually in humans it should work, but if the same drug is given to person who is already taking some other drugs, or is diabetic, or had blood pressure, or all those, उसके enzymes, उसका tension, उसका, क्योंकि अगर diabetic है तो glucose levels, insulin levels are different. Those may have, उनके enzymes are working slightly differently, and that has to be balanced with the drug for which intended drug is, right? So sometimes if those comorbidity of some other reason is happen, then the physician try to adjust that कि आपने कब कब लेना है, कौन सी दवाई के साथ में दूसरी दवाई लेनी है या नहीं लेनी, because drug interaction, the pharmacokinetics of the one drug should not negatively influence the pharmacokinetic of the second drug. If it influences, उसका असर नहीं होगा. And then many times when you see, you go to the clinician कहता है कि ठीक है इस दवाई को इतने बजे लेना, फिर इतना गैप रखो, फिर दूसरी दवाई को इस समय लेना. So इसमें when next time when you hear that, घबराने की जरूरत नहीं है. Now as a chemist you can explain कि वो क्यों ऐसा कह रहा है, right? कि उसे कहने का मतलब क्या है, क्योंकि otherwise if you do not know that, बोले कि डॉक्टर कह रहे हो से मैंने मालूम नहीं क्यों कह रहे हो से, राइट? तो घबराने की जरूरत नहीं है. नाउ एज ए केमिस्ट यू कैन एक्सप्लेन व्हाई इट इज हैपनिंग, क्योंकि वो ड्रग का आपस में इंटरेक्शन ना हो और जो दोनों ड्रग का असर हो, सिर्फ इसीलिए वो कह रहे हैं. उसको भी ज़्यादा, दैट्स व्हाट दैट डॉक्टर इज ट्राइंग टू बैलेंस. सो एज ए केमिस्ट यू कैन सी देयर इज लॉट मोर अप्रिशिएशन. अब ड्रग या ये सब चीज़ों को सुनो, डर के भागने की जरूरत नहीं है. यू जस्ट ओनली कैन नो आई टोटली अंडरस्टैंड कि ये सब क्यों किया जा रहा है. इट्स सिंपली प्रॉपर्टीज, ड्रग की प्रॉपर्टीज आई एम ट्राइंग टू मैच एंड ट्राई टू एक्सप्लेन बॉडी के अंदर ही किस तरह से कौन सा ड्रग कब लेना है, क्यों लेना है, कितना लेना है, कितनी बार लेना है और कितने अल्टरनेट रूट पे लेना है. इट्स ऑल कम्स डाउन टू एडमी प्रॉपर्टीज. दैट्स ऑल देयर इज. Okay, so far look at, I'm emphasizing, I made my slides specifically keeping chemistry in focus because most of you, if not all of you, have done more chemistry. But now look at that, it's all comes down to what is the molecular weight, how many hydrogen bond, is it lipophilic or lipophoric, what is the pHa, this is polar surface area, PSA, right? What is the pK, whether it's an acidic or basic when it breaks down to, right? Uski reactivity kya hai, shape, when I say shape, three-dimensional shape, because if you have a chiral center, right, you have two isoforms, उसकी shape क्या है, वो काम करता है नहीं करता है, और वो इतना अंदर जाके stable हो गए. As you can see, all of these are nothing but only chemistry. All I talked is just chemistry. Because this is based of that is उसके physical, किस physical environment में जा रहा है. This is what I talk about. When I say physical environment, कौन से enzymes उसको interact करने वाले हैं, वो body उस path के अंदर, कौन कौन से proteins को वो target करने को जा रहा है, right? मगर देखो journey यहाँ से शुरू होती है. It starts from here. So if it is not working, you bring back, you fix that. If it is not working, you bring back, you fix that. You keep on modifying, keep on optimizing. That is what is called hit to lead optimization. कि आपको initially जो hit मिले, आप उसको optimize कर रहे हैं. That's a very, assuming, have you guys heard the term hit to lead optimization in a drug? आपको initial hit मिले, आपने 10,000 कंपाउंड स्क्रीन किया, आपको 200 मिले. वो जो मैंने आपको यहाँ, I showed you a picture, right? जो टारगेट दिखाया था कि वो जो एरो है ऊपर है, कि इनिशियली वो कुछ काम कर रहा है, अब आप उसको ऑप्टिमाइज कर रहे हैं इन सब चीज़ों को ध्यान रखते हुए. एंड देन बेंग टू बुल साइज, एकदम सेंटर में लेके आने का. दैट्स बेसिकली ऑल. एंड देन क्लियरेंस पाथ क्या है, उसकी हाफ लाइफ कितनी है, बिकॉज दैट हेल्प्स टू डिटरमाइन डोज कितना लेना, कितनी गोलियां लेनी, दिन में कितनी बार लेनी या नहीं लेनी, अल्टरनेट पे लेना. बायो अवेलेबल, कितना वो अवेलेबल है, व्हाट इज द क्लियरेंस हाफ लाइफ. अगर 5 मेग लिया है और 5 मेग के अंदर उसका कई है, उसकी फिजियोकेमिकल प्रॉपर्टीज, ड्रग का स्ट्रक्चर ऐसा है कि वो धीरे से रिजॉल्व होता है, मतलब कि वो प्लाज्मा और बॉडी में लॉन्ग टाइम तक रहेगा. सो यू वी टेक वंस अ डे, वो रहेगा उसमें धीरे धीरे रहेगा. देयर आर सम ड्रग्स, उसका कई ड्रग जो देते हैं उसका सिर्फ 30 मिनट, 60 मिनट, 90 मिनट ही होता है. वेरी चैलेंजिंग, बट दैट्स वेरी मोस्ट इफेक्टिव. देन डॉक्टर विल ट्राई टू फिगर आउट क्विकली उसको कैसे देना है. IV दो, फटाफट पहुंचा, उसने अपना काम किया, बजाय इसके कि पिल्स दी और आपको जी हर 1 घंटे में एक पिल लो. दैट्स नॉट प्रैक्टिकली पॉसिबल, राइट? सो दैट्स हाउ दीज थिंग्स आर. सो उसकी अगेन लीथल डोज क्या है, टॉक्सिसिटी, कि कितना जो डोज, जो लीथल डोज है उससे नीचे रखने का उसकी कॉन्सेंट्रेशन, क्योंकि उससे हाई कॉन्सेंट्रेशन पहुंचा, it will have some lethality, it will have some negative effect on it. So just to avoid, so see here, it's all chemistry, but actually it's drug discovery, it's drug development, it's making medicine, it's making therapies, it's making drugs. Any question? You want to take a break? Okay, let's take a few minutes break and then we'll come back. Right, so let's continue with the pharmacokinetics and me talks. So this is again just to summarize what we've been talking. Okay. So again, this is again, I already explained to you, this is just to get started and see where, and here I have listed only few enzymes and proteins which play a role. किसी भी चीज़ के लिए absorb करने के लिए केको is a test, is a thin layer होती है केको की between the cells. It has to permeate. So जब टेस्ट करते हैं उसमें केको टेस्ट किया जाता है. When we test, उसमें केको लेयर बनाई जाती है cells की, and then you take your drug, you dissolve in a whichever solvent you have, and then you see वो permeate करके दूसरी तरफ जाता है या नहीं जाता drug उस chamber के अंदर. So it tells you whether it will cross distribution, the drug will pass on. So that's a Keiko test for absorption, whether it will absorb or not. Same way, these are various enzymes जिसके test बने हुए करने को. Distribution again in the plasma, what drugs, whether it's absorbed, whether it will bind to the plasma protein or not, कितना volume होगा, blood-brain barrier. I think I mentioned yesterday or day before. If the drug which is needed to pass for brain, अगर वो उसको cross नहीं करता तो drug is not going to be effective. It can be effective other places. So you have these tests. So when we do these tests, for example, in my lab or any lab that does drug discovery, drug development, when you try to see if it has to get to the brain, it has to cross to the BBB, blood-brain barrier. It has to pass. So ये various tests बने हुए हैं जो किए जाते हैं. ERG toxicity is the human ether agogo related gene. Very commonly you hear this ERG toxicity for cardiotoxicity, heart के लिए. कि उसको जो ERG की toxicity, that's an enzyme which is extremely important for cardio functions. And अगर वो cardiotoxic है drug, तो ऐसा नहीं कि अगर drug वो कैंसर पे काम कर रहा है मगर वो उसके कारण heart का असर हो रहा है. And there are drugs actually. So we are working on many of these drugs to optimize those drugs, which are drugs actually in the market. They are highly effective. For example, there is a drug as I mentioned yesterday, one of the targeted therapy started from my institute. The very first drug came out. Uske baad mein now there are 74 drugs in the same class which are in the market. But one of the recent drug is known as ponatinib. Highly potent, बहुत effective है at a picomolar concentration and single digit nanomolar concentration. But it is highly toxic, cardiotoxic. So drug was withdrawn कि वो असर तो कर रहा है but उसके कारण इतना cardiotoxicity, heart problem हो रही थी patients को, तो drug was withdrawn from the market. So ERG is, for example, is one of the things which is tested. So this is LD50, as I showed previous slides, maybe lethal dose 50% that causes the acute toxicity. वो डोज उससे नीचे होना चाहिए. अगर उस डोज पे है जो 50% toxicity करता है, then that's not the right dose. So all these doses, the CYP is an enzyme. वो CYP को inhibition कितना होता है, metabolite वो break down करता है, liver enzyme है. So just to recap, all these, now you can slowly, hopefully I'm assuming that you have an appreciation of what it takes, कितने disciplines involved है एक drug को market में लेके आने के लिए, कितनी expertise चाहिए, कितनी सब specialty चाहिए, एक चीज़ समझ आनी जरूरी है in order to see. And this is again a very simplified version of how many different things which are studied before drugs are developed. So basically, idea is you are juggling many balls in the air, and you're trying to keep each ball in the air. And that is the property. You're juggling multiple different aspects. ADME ठीक होना चाहिए, properties ठीक होना चाहिए, distribution ठीक होना चाहिए, effect ठीक होना चाहिए. सब कुछ को balance करते हुए आगे लेके जाना है. And as I showed earlier, as you can see now, जो मैं भी थोड़ी देर पहले कह रहा था कि एक ड्रग को वहाँ से, एक कैंडिडेट को वहाँ से वहाँ पहुँचने के लिए कितने जो हैं चक्रव्यूह में से निकलना पड़ता है, कितनी कसौटी से निकलना पड़ता है to be successful, to reach to the point of, to the site of action to become a drug. So देखिए कितनी जो हैं, all these tests have to be passed for a drug to become a drug. And so that is why design of a structure, design of a drug is extremely, well, depending on if you like, it's a lot of creative fun exercise when you are thinking of many properties, many different aspects, and you are just working on that chemical structure, even a small molecule case, in order to see कि वो आपका इस तरह से हो कि आपको वो सारे जो चक्र व्यू हैं उनमें से पास हो कि आपका ड्रग जो है एक्चुअल ड्रग बन जाए. So this is again iterative process. This is what I was trying to say, that you have to in vitro studies पहले outside the any in vivo animal study के बाहर in vitro petri dish में studies वो पास होता है. Then you have to have pharmacology. अगर pharmacology काम नहीं करिए फिर से synthesis कीजिए, फिर काम नहीं करिए फिर optimize करिए उसको. So it is a very iterative process, again and again process, up until you are satisfied that all these properties are meeting. When all these are satisfied in vitro, not going into the animal body, that's when you take inside the in vivo work. And that's when this whole process of understanding and the in vivo performance. So in vitro properties, सारे rules उसको जो भी properties के पता हैं, करते कराते you bring it here, do in vivo test. Do in vivo test, and again it has to pass through. So just imagine, just for the ADME, just for the optimization, how many different, and here, how many different things are taken into consideration. कि आपका मॉडल ठीक होना चाहिए, आपका एसिड करेक्ट होना चाहिए. ऐसे नहीं कि आपने अपनी तरफ से तो ऐसे बना दिया कि बहुत अच्छा है मेरे को बताता है, बट इफ इट हैज़ नो रेलेवेंस कि बॉडी के अंदर क्या हो रहा है, तो आपका ऐसे इज जस्ट एन ऐसे, इट्स नॉट गाइडिंग यू, it is not guiding you to design the drug. So see how many different things are taken into consideration: absorption, metabolism, distribution, brain exposure, dosing, bioassay, toxicity, just a few of those. In सब का ध्यान रखते हुए that your drug has to meet all these conditions. And then now based of, again there is just a few examples of various molecules just based on their lipophilicity. वो कितने हैं ऑर्गेनिक हैं वर्सेस नॉन ऑर्गेनिक हैं, उनकी सॉल्युबिलिटी क्या है, लिपोफिलिसिटी क्या है. एज यू कैन सी हियर दे फॉल एट, उनकी लॉग पी वैल्यू क्या है. सी मॉर्फिन कहाँ पे है ऑन द लॉग पी वैल्यू इसके ऊपर, एंड देन वेदर दे क्रॉस द बॉर्डर और नॉट. ये vinblastine is a cancer drug, vincristine is a cancer drug, morphine as we were talking earlier is a painkiller, and many other things for sedative it is given. But उन सब का जो है असर L-DOPA for depression drug brain के लिए दिया जाता है. And so polar surface area उसका क्या है, उसकी log P polarity उस drug की क्या है. Based of that, in properties के basis पे with the oral drug और IV drug कितने टाइम देना है, क्या देना है. So hopefully with all these you have an appreciation and understanding कि what it takes कि एक drug को करने के लिए. But again, as I emphasize again and again, as a chemist, as an organic chemist, you have lots and lots of advantage because you can understand the properties, you can design the properties, you can manipulate the properties. So far what I've been telling you is, you know, the things which are the properties taken into consideration important, but again in vivo and in vitro में क्या क्या किया जाता है, which is quote unquote preclinical part. कि preclinically before it gets to the clinic, right? So pharmaceutical industries जो करती हैं, मेजर बायोटेक फार्मास्यूटिकल इंडस्ट्री जो करती हैं, उनका सारा इन्वेस्टमेंट, टाइम, एफर्ट, रिसोर्सेज इज टू डू ऑल दीज स्टडीज बिफोर इट गेट्स टू क्लिनिक. तो जितनी भी बड़ी बड़ी इंडस्ट्रीज हैं उनकी सारी इन्वेस्टमेंट जो है उसमें होती है कि एक ड्रग को कैसे निकालना. और नाउ यू कैन अंडरस्टैंड कि उसमें कितने सारे सब्जेक्ट्स, कितने सारे डिसिप्लिन इन्वॉल्व हैं. बट वही वो सब करते हैं. दैट्स वाई इट इज़ अ वेरी एक्सपेंसिव प्रोसेस बिकॉज यू हैव टू हायर दैट मैनी पीपल, दैट मैनी एक्सपर्ट्स. वो अपना पूरा ब्रेन पावर लगाते रहेंगे इंडिविजुअली, कलेक्टिवली, टू come with a candidate, come with a drug that can go to the clinical trial. So this is, you know, preclinical study, दो टाइप जो मैंने आपको अब तक के लिए बताया. But then again, ultimate goal for any of these things is to get to the clinic, right? जो ये सब कर रहे हैं पेपर छापने के लिए तो सिर्फ कर नहीं रहे, हमें भी हो सकता है करें, but ultimate goal is to make it into, convert it into a drug which can help other humans. So clinical trials जरूरी हैं. So preclinically जो हम जितने करते हैं, yesterday there was a question and I think a couple of times question asked के why when we are testing in animals will it really work in humans? So answer is we don't know. But आप ह्यूमंस को तो गिनी पिग तो नहीं बना सकते ना कि भाई चलो टॉक्सिसिटी कोई बात नहीं बेचारा मर गया, वो तो काम नहीं चलेगा ना. आपको पहले टेस्ट करना है एंड देन देयर बॉडी के जो मेकअप है इट इज़ नॉट ऑब्वियसली नॉट आइडेंटिकल एज ह्यूमंस, नहीं तो वो भी ह्यूमंस होते, बट एज बेस्ट एज वी नो कि उनका जो मैकेनिज्म है इज क्लोज टू व्हाट वी सी इन द ह्यूमन. सो दोज आर द मॉडल्स विच आर कलेक्टेड. माइस में स्टडी की जाती है इसलिए नहीं कि माइस इज सिमिलर टू ह्यूमंस, because those are relatively cheaper. It's a very expensive process. Mice breeding, I don't know यहाँ पे breeding को या किसी भी डिपार्टमेंट पे यहाँ पे होती है, बायोकेमिस्ट्री, बायोलॉजी या वेटनरी यहाँ पे स्कूल है या वेटनरी, बट ब्रीडिंग जो है अदर डिपार्टमेंट्स यहाँ पे इंडिया हो ब्रॉड हो वो अपनी माइस ब्रीडिंग खुद करते हैं बिकॉज इट्स रिलेटिवली ईजियर टू डू. इफ यू डू द सेम स्टडी इन पिग्स or in even mice or rats में जाओ they are expensive. Then you going to pigs or जो rabbits they are more expensive. And to taking it to dogs and for monkeys it's like एक monkey study, one of the largest monkey colony in the US is in my university. half a billion dollar का उनका जो सिर्फ research work है comes from industries. इंडस्ट्री जो करते हैं इंडस्ट्रीज के पास में तो ये सब है नहीं क्योंकि उनको जरूरत नहीं है एक ड्रग के लिए इतना पूरा मंकी कॉलोनी रखें. बट देन इट्स द ओल्डेस्ट वन विच वाज सेट अप बाय जापानीज ग्रुप इन 50s में एंड देन यूएस गवर्नमेंट टुक इट ओवर एंड नाउ इट्स बीइंग रन. बट पॉइंट बीइंग उसके लिए उसका मॉडल को बनाना तो जितने कई जो जोड़े ड्रग है एक्सपेंसिव ड्रग्स हैं व्हाटएवर दोस ड्रग्स आर व्हिच आर इन द मार्केट व्हेन दे कम देयर उसके लिए जो मंकी स्टडीज़ many of these studies happen in our universities for companies क्योंकि उसका जो मॉडल बनाना ठीक मॉडल बनाना मंकी की क्या एज होनी चाहिए जिस पे वो टेस्ट किया जाए जो कि रेलेवेंट टू व्हाट इज़ द ह्यूमन ट्रांसलेट करने के लिए उसका इफेक्ट होगा. जस्ट सिंगल मॉडल कुड बी अबाउट 250 to 300,000 US dollars जो कि 2 से 3 करोड़ एक मंकी स्टडी करने के लिए. So just imagine nobody can afford that up until it gets to that point. So उसके लिए कराने के लिए this is a very step-wise, very tedious and highly expensive process. So इसीलिए जो criteria बने हुए कि कैसे उसको filter कर दो कि पहले ही पता चल जाए काम करेगा कि नहीं करेगा, तो उसके लिए बड़े सारे in vitro test बने हुए हैं, in vivo test बने हैं simplified way में. So you know that whether it's going to work and then you slowly gradually. So agencies like Food and Drug Administration जो US में है जो रेगुलेशन जो अप्रूवल देता है ड्रग के लिए उनका क्राइटेरिया है कि इन मैनी केसेस यू हैव टू शो इन 2 स्पीसीज, 2 डिफरेंट स्पीसीज के अंदर आपने अपने ड्रग की सारी स्टडी शो करनी होती है. एक जैसे माइस है, रैबिट हो गया, या कुछ केसेस में डिपेंडिंग ऑन व्हाट ड्रग एंड व्हाट डिजीज इज यू हैव टू शो इट इन मंकी और डॉग्स. एक एक को करने के लिए यू कैन जस्ट इमेजिन एक एक करना स्टडी करने तो 2 से 3 करोड़ रुपए सिर्फ एक को करने के लिए चाहिए. एंड देन इफ यू हैव टू डू इन फ्यू टू टेस्ट दैट. सो दैट इज हाउ दिस ग्रेजुअल पाथ उसका बना हुआ है. एंड इट्स नॉट कि सीधा क्योंकि ह्यूमन में सीधा क्योंकि उसमें क्यों नहीं जाते बिकॉज इट्स अ वेरी एक्सपेंसिव प्रोसेस. इट्स एक्सट्रीमली एक्सपेंसिव प्रोसेस टू ड्रग डिस्कवरी. सो एनीवे सो अल्टीमेट गोल इज एज वी बीन टॉकिंग इट्स नॉट कि आपने इन विट्रो करके देख लिया और सीधा ह्यूमंस में ले जाओ. इट्स नॉट पॉसिबल क्योंकि इन विट्रो में तो आपको देगा क्योंकि कंडीशन यह है कि आपने जस्ट थिंक फॉर अ सेकंड इफ यू डेवलप अ ड्रग the idea is to the drug has to be effective inside human body. If you take those enzymes and put it in a petri dish, वो सिर्फ एक अकेला एंजाइम है, एक सेल है. ह्यूमन बॉडी जो है उसके अंदर कुदरत ने बहुत सी चीज़ें और डाली हुई हैं. आप उसको डुप्लीकेट तो कर नहीं सकते इतना आसानी से बॉडी के बाहर. राइट. सो अभी जैसे कई मॉडल आ गए हैं, और हैव यू हर्ड द टर्म ऑर्गेनॉइड्स, spheroids and organoids? So 3D models you can create inside. Those are 3D models. PDX, patient derived xenograft model, which is, so initially, previously what, and even now continues to happen, is you have to do in vitro study. Petri dish के अंदर आपने एंजाइम ले लिए, प्रोटीन ले लिए, उसका टेस्ट कर दिया. That's the quickest and easiest way. चलो आपको पता चला काम करता है या नहीं करता है. आप उसको आगे लेके जाना है 3 डायमेंशन के अंदर. So you have organoid models और xenograph model अपने माइस में किया. अब माइस में भी किया कि डजंट मीन जो माइस में किया, माइस आर नॉट ह्यूमन, तो उसकी जो फिजियोलॉजी है इज नॉट सेम एज द ह्यूमन फिजियोलॉजी, राइट. सो देन इन पास्ट 15 years or so, there is another model, type of model developed, which is called PDX, patient derived xenograft model. In that case, what happens is you take a sample of, for example, let's say in case of cancer, triple negative breast cancer or breast cancer, for example, you take a piece of the patient sample, you engraft inside the mice and you let it grow. उसमें से लेके second passage या then you put into the next generation. Idea is कि by that जो tissue है is inside the xenograph के अंदर, वो उसके अंदर आ जाए. So now it's in 3 dimension. Then you study your drug in there. So it is not human, but it is one step closer to what you can see. But उसमें भी एक पूरी अपने आप में field है, क्योंकि बहुत जल्दी कर दिया तो माइस के सेल्स होंगे, बहुत लेट किया तो भी माइस के सेल्स होंगे, क्योंकि आफ्टर सम टाइम द होस्ट विल टेक ओवर द गेस्ट सेल्स. तो उसकी भी अपने आप में फील्ड है कि कितने पसाज पे उसको करना नहीं करना. बट यू सी दैट्स मॉडल टू क्रिएट द राइट मॉडल्स, ऑर्गेनाइज्ड मॉडल्स, एक्स वीवो ह्यूमन मॉडल्स. एक्स वीवो ह्यूमन बॉडी के बाहर है, बट वो ह्यूमनाइज्ड मॉडल्स हैं जो बनाए गए, उनमें टेस्ट करना. सो यू आर स्लोली उसके अंदर फिल्टर डालते जा रहे हैं कि आपका ड्रग पास कर रहा है, वो टेस्ट को पास कर रहा है, तब धीरे धीरे उसको आगे लेके जाते हैं. एंड इवन आफ्टर दैट इट इज स्टिल नॉट ह्यूमंस, यू डोंट नो वेदर इज गोइंग टू वर्क और नॉट, बट यू होप. बट दैट्स व्हाई इतने सारे उसमें चेक्स एंड बैलेंसेस हैं कि किसी भी स्टेज पे अगर नहीं काम करता तो जरूरी नहीं है कि सीधा ह्यूमन पे ले जाओ. यू डोंट वांट टू, यू नो, और उसके एक तो बहुत एक्सपेंसिव है. इफ इट इज नॉट गोइंग टू वर्क, then what is the point of taking it to the humans, right? Or most importantly, the safety. अगर वो safe नहीं है तो you cannot just use human as a guinea pig और टेस्ट करना शुरू करो. So that's why all these developing right model, choosing right model, किस डिजीज के लिए आप कर रहे हैं वो मॉडल उसके लिए काम करता भी है नहीं. ऐसा नहीं कि पिग में किया तो सबको पिग में कर दिया और मंकी में किया तो सबको मंकी में कर दिया. किस डिजीज के लिए, किस काम के लिए, whether it is relevant or not, that understanding. So as you can see, the reason I'm mentioning all of these things is so you have an appreciation of what really it takes. So that's what basically experimental therapeutics is. It's a field which is multidisciplinary. You bring multidisciplinary, but as a chemist, again, I'm emphasizing again and again, as a chemist, you can look from the lens of chemistry into all these pieces step by step. Before you know, you become into a medicinal chemist or you become like a toxicologist, pharmacologist, you become all of that. But many of the leading pharmacologists, toxicology, ADME wale log, PD wale log, they are trained chemists in the world. They are trained chemists because they look from the chemistry lens. They understand कि buffer है तो buffer की solution की properties क्या होगी, human plasma है उसकी properties क्या होगी, आपके drug की properties क्या होगी. But that's how these things are done. Models वगैरह हों. So in next few slides now I'll just emphasize on the clinical side कि क्या है, just in vitro or in vivo preclinical part. So if you look at, as I mentioned about dose, remember जो dose के बारे में अगर बात करें तो look here कि कितनी चीज़ें इन्वॉल्व हैं. एब्जॉर्प्शन, प्लाज्मा में गया कितना गया, डिस्ट्रीब्यूट हुआ या मेटाबोलाइट होके बाहर हो गया वो. So I'll give you an example of. So we were working few years ago, we were working on a drug जो यूरोप में अप्रूव था, यूएस में अप्रूव नहीं था. So we were interested in trying to see to bring it to for multiple sclerosis. उसके लिए, ड्रग के लिए. अब उस ड्रग का चैलेंज ये था कि मल्टीपल स्क्लेरोसिस के अंदर जब हम उसे टेस्ट करने की कोशिश कर रहे थे, ड्रग इज सो हाईली सॉल्युबल कि 97% वो क्लियर हो जाता ड्रग फटाफट. ड्रग आपके इन विट्रो में बहुत अच्छा काम करता था, इन वीवो में उसका बहुत ज़्यादा हैवी डोज देके करो तो उसका असर नज़र आता था. बट वो 97% वो मेटाबोलाइट ना क्लियर हो गया, बाहर हो गया. ओनली 3% उसके अंदर बचा. तो आप उसको बकेट लोड दोगे थोड़ा सा असर करने के लिए, दैट्स नॉट द वे टू डू इट. सो वी एज ए केमिस्ट लुक एट द स्ट्रक्चर कि उसके थोड़ा सा उसके प्रॉपर्टी को कैसा चेंज करें कि उसका जो एक्टिव पार्ट है वो मेंटेन रहे मगर उसकी स्टेबिलिटी ज़्यादा रहे. वो प्लाज्मा के अंदर सिर्फ आधे घंटे में 97% निकलने के बजाय वो कम से कम 6-8 घंटे तक प्लाज्मा के अंदर रहे. All we did was we designed a lot of analogs around that and then started testing those analogs. Now the other challenge of that drug was, वो एग्री करते हैं, the other challenge of that drug was कि वो ड्रग इतना उसकी लिपोफिलिसिटी, जैसे मैं आपको अभी I showed you the property lipophilicity. The other thing about that drug was कि वो ड्रग जो है इतनी जल्दी से मिल जाता था कि उसको टेस्ट करने की कोशिश करो, किसी भी वो क्रोमेटोग्राफ किसी में निकलता ही नहीं था, वो सेल के अंदर कहीं चिपक जाता था. तो आपको पता ही नहीं, आप देखने की कोशिश कहीं पे यू डोंट सी एनी पीक. दैट्स ऑल यू कैन डू. अब वो पीक के अंदर पड़ गया, ड्रग असर कर रहा है, विच मीन्स समथिंग इट इज इनसाइड, बट यू कैन नॉट फॉलो द कॉन्सेंट्रेशन के कितना अंदर गया कितना बाहर गया. तो देन एज ए केमिस्ट यू गो बैक अगेन स्लाइटली, उसका सॉल्ट बनाएं या चेंज द प्रॉपर्टी और चेंज उसका जो फॉर्मुलेशन उसको चेंज करो, बट अगेन ऑल केमिस्ट्री. सो वी डिड ऑल ऑफ दैट. So drug which was in Europe, approved in Europe, we brought the drug to multiple sclerosis. We made changes to the drug और उसका जो क्लियरेंस 97% निकल जाता था आधे घंटे में ड्रग बाहर, उसका एनालॉग्स बना के चेंज किया कि वो 6-8 घंटे तक प्लाज्मा में रहता था. And then when you study, we saw that our new candidate was much more effective than the original drug. But I'm just to give you an idea कि how these things become important. So अगर वो जल्दी eliminate हो गया, again go back, design the drug, because and again depending on where the action is, if it is something to do with brain, neurological कुछ देना है, it has to cross through the blood-brain barrier. अगर वो पास ही नहीं होगा तो कोई फर्क नहीं होना, उसका ब्रेन पे असर नहीं होना. So you again try to play with the property. This is again in the process, you know, इतने stages होते हैं ना, as I mentioned, कि ड्रग आगे जाएगा या नहीं जाएगा. There are many go no go decisions. So in the pharma industry, हमेशा they are working on इसको आगे लेके जाएं या नहीं लेके जाएं. But just imagine, it's a huge investment. वो किसी गलत ड्रग को आगे लेके जा रहे हैं, कितने लोग इन्वॉल्व हैं, उन सबकी सैलरीज, कितने एसेस, कितने मॉडल, बिफोर यू नो वो करोड़ों में आ जाता है. अगर उसकी प्रॉपर्टीज मैच नहीं करती, देन उनको डिसीजन लेना पड़ता है कि स्टॉप हियर, लेट्स फर्स्ट चेंज इट. सो दोज गो नो गो डिसीजंस आर द फार्मा इंडस्ट्री के अंदर जो साइंटिस्ट हैं, उनका ब्रेन इज ट्यून टू गो नो गो डिसीजंस बिकॉज इट्स अ वेरी एक्सपेंसिव एंटरप्राइज. सो बट सी how many different aspects that play into role. This is a simplified version. Look at this for clinical development. This is a very simplified version again. A simple flow chart कि अगर आपने ड्रग लिया है यहाँ पे तो जाके okay, now you reach to the point कि सारे प्री क्लिनिकल जो हैं आपके they are good. You agree कि जितने प्री क्लिनिकल मॉडल्स इतने थे, when I say preclinical including in vivo model, animal models, they give satisfactory results. Okay, this is fine, now let's take it forward. Now you enter into phase 0 or phase 1 clinical trial. You enter clinical trial, you bring it to this point and see in the phase 1, usse pehle I'll bring back one of my lecture tomorrow कि clinical trials के अंदर approval क्या लेने पड़ते हैं, किस तरह से लेने पड़ते हैं. But again, these are various decision point. आपने so phase 1 phase 2 की क्या difference, I'll bring that tomorrow, I'll not mix it right now. But point what I'm trying to say is for clinical development के अंदर again there are so many steps involved after in vivo काम हो गया है, क्योंकि अब now you are entering into the humans के अंदर. You have to be extremely careful कि किस चीज को क्या टेस्ट करना है and how much to take forward or not to take forward. So this is just to give an, again to emphasize the relationship between dose and the effect. So dose जो दिया गया है, whether again it is reaching to the site of intention action or not. If it is not, then you have to change the dose, you have to change the formulation, you have to change the route of administration, oral, dermal, IV, you change so it reaches. So again, as you see here, same point. मैं क्या dose कुछ heavy हो गया था. I don't know, is it still raining outside? Maybe because of. Is everybody inside? Sabanda? Okay. Okay, so we'll try to keep the dose low so the electricity is not affected. So as you see here, how dose and effect are all taken into consideration, and this is just to give you another. It's not moving forward. Okay, so how, as I was telling you पहले कि आप किस डोज से देते हैं, वो किस तरह से पहुंचता है आपके साइट पे, उसका डिस्ट्रीब्यूशन किस तरह से होता है, इट वेरीज. सो सी हियर फॉर एग्जांपल, यू नो देयर आर वेरियस रूट्स. आप अगर आईवी देते हैं, दैट्स द फास्टेस्ट. ये जो है 100% पहुंच जाता है, क्योंकि आईवी में दिया ब्लड उसके सर्कुलेशन में इमीडिएटली पहुंच गया वो साइट पे, डिस्ट्रीब्यूशन जो करना है. अगर उसी चीज़ को इंट्रामस्कुलर दिया, मसल्स के अंदर इंजेक्शंस दिया, तो अबाउट 75% जो है उसका डिस्ट्रीब्यूशन फास्टर होता है. इफ़ सब क्यू दिया या ओरल डोज दिया, depending on the property of the drug, it could be very small percentage that gets to the site, or it could be 100% if the drug की property ऐसी है कि it survives through all the things that I was telling you earlier. If it survives, उनमें बचा रहता है, चाहे pH है, जो enzymes हैं उसमें survive करता है, तो वहाँ तक वो पहुँचेगा. So depending on which, you know, ये rectal चाहे आजकल जैसे inhaler आ गए हैं, you inhale the drug, right? Nano applications वगैरह आ गए, you inhale the drug. आपने देखा है जो विक्स का वो आता है फॉर एग्जांपल, दैट्स इनहेलर. इनहेल करने से जो है उसकी ऑब्वियसली इट इज़ गोइंग टू योर नोजल प्रोसेस, नोजल पाथ के थ्रू इट इज़ ट्राइंग टू गेट देयर. उसमें से कितना % वो पहुंचेगा, कहाँ तक पहुंचेगा, उस इट में वेरी. इट कुड बी वेरी स्मॉल डिपेंडिंग ऑन पर्सन, इट कुड बी ऑल ऑफ इट कैन गो. और ट्रांसडर्मल, स्किन के नीचे या आपने जो लोशन लगाते हैं वो कहाँ तक पहुँचते हैं. सो कितना अवेलेबल, बायो अवेलेबिलिटी उसकी क्या है, वो कितना एक्चुअल फॉर्म में अवेलेबल है, कितना नहीं अवेलेबल, इट हैज़ क्लियर. उसका इफेक्ट जो है ऑब्वियसली टाइम लगेगा उसको, क्योंकि साइट पे पहुंचेगा, अपना असर दिखाएगा. वो डिपेंड करता है कौन से रूट के ऊपर है. सो देखिए आईवी एज एवर से कि इट इज़ रेडिली अवेलेबल, क्योंकि प्लाज्मा में जाता है फटाफट डिस्ट्रीब्यूट होता है. आईवी में जब देते हैं नर्व के अंदर ना लगाते हैं तो सीधा फटाफट डिस्ट्रीब्यूट जल्दी होता है वो, तो उसका असर जल्दी होता है और लो कॉन्सेंट्रेशन पे उसका असर होता है. इसी तरह से वॉल्यूम, ये सब क्यों दें. ओरल का मोस्टली कोशिश की जाती है कि ड्रग्स जो है ओरल फॉर्म में कन्वर्ट किए जा सके, क्योंकि एक पिल लेना बड़ा आसान है. लिया आपने एक लिक्विड वगैरह के साथ में लिया, बड़ा आसानी से ले लिया, बगैर इसके कि आपको वो आईवी लो. आईवी के लिए आपको हॉस्पिटल में जाओ या इंजेक्शन दो और वो सब करो. इट बिकम्स मोर चैलेंजिंग एंड नॉट नेसेसरली कि जो एवरीबडी विल एक्सेप्ट दैट, है ना, कि चाहे वो सिंपल जल्दी फैम बढ़ जाएगा. आपने जरूर देखा होगा, कहीं भी इसमें करते भी होंगे, द मोमेंट डॉक्टर्स भी कहीं हैं, जैसे वो इंजेक्शन को देते हैं, वो जो डॉक्टर स्टूडेंट जैसे इंजेक्शन देखते हैं वो फेंट करके खुद गिर जाते हैं. यू नो नॉट, बट इट्स ओनली द प्रोसेस देने का है, राइट. सो नॉट एवरीबडी इज सो ईजिएस्ट एंड द मोस्ट वे इसके, अगर डर्मल है, किसी को डर्मल दिया, जैसे आपने क्रीम लगाई, हो गया काम, इफ़ उस चीज़ के लिए. अदरवाइज मोस्टली कंपनियां कोशिश करती हैं, जो भी ड्रग डेवलपर कोशिश करते हैं कि उसके ओरल राउट उसका दे सके, कि आपने पिल लिया, पानी पिया, हो गया, अपने आप डिस्ट्रीब्यूट होता रहेगा. सो दैट्स अगेन दैट्स ए बिजनेस डिसीजन, प्रैक्टिकल डिसीजन कि आप दें. अगर कैंडिडेट ले भी तो सही, जो पेशेंट ले भी तो सही, अगर लेगा नहीं और उसको अवॉइड करेगा, उसको कोई भी हिकअप्स हैं, कोई भी उसको जो है रुकावट है सोच में, वो ना रहे, तो ओरल्स आर रिलेटिवली कंसिडर इजियर. बट उसका चैलेंजेस ये हैं कि अवेलेबिलिटी जो है इट वेरीज, एंड दैट्स द रीजन नाउ यू सी. क्योंकि अगर सेम ड्रग है, अगर आपने आईवी देना है तो हर बार पेशेंट को आना पड़ेगा, आईवी देना पड़ेगा, राइट. आईवी देने के लिए एक तो वो एफर्ट है कि आप घर से आएगा, आईवी देगा, उसके लिए पूरी मशीनरी चाहिए, आपका पूरा स्टाफ चाहिए, फिजिशियन चाहिए. एंड नाउ जस्ट इमेजिन कि वही ड्रग जो उसने देना है, अगर चाहे इंश्योरेंस कंपनी दे या अपनी पॉकेट से जो भी दे, कितने नर्सेज इन्वॉल्व हैं, क्लिनिकल कोऑर्डिनेटर इन्वॉल्व, फिजिशियन, उन सब की सैलरीज आ गई, इसीलिए उसका बिल बड़ा लंबा आएगा. बजाय इसके अगर ओरल है, उसने खुद ही लिया, काम हो गया. सो these are the business decisions that are taken into consideration. And again, it goes back to, as you see here, कि उसका डिस्ट्रीब्यूशन है वो कब जो है उसकी कॉन्सेंट्रेशन क्या होगी ब्लड के अंदर और कितना टाइम लगेगा उसको. ड्रग रैपिडली कंप्लीटली अवेलेबल, ड्रग विच आर ओरल हाफ अवेलेबिलिटी. क्या अवेलेबिलिटी? द थिंग्स विच आई हैव बीन टेलिंग यू. ड्रग कंप्लीटली अवेलेबल बट रेट ओनली हाफ ऑफ ए. जो फुल्ली अवेलेबल है उसके मुकाबले में थोड़ा कम अवेलेबल है. सो ये सारे पॉइंट्स आर टेकन इनटू कंसिडरेशन एक डिज़ाइन में. और जो एक तो पहला है प्योरली साइंटिफिक एट द मॉलिकुलर लेवल अंडरस्टैंडिंग कि उसका स्ट्रक्चर क्या है, उसकी एंजाइम्स किस साथ में इंटरेक्ट करेगा, सारा वो साइंटिफिक पार्ट को समझना. उसको समझने के बाद में देन इट कम्स द एप्लीकेशन पार्ट कि अगर आप वो दे रहे हैं तो किस तरह से किस रूट से दिया जाए उस ड्रग को, उसके कारण उसकी कंप्लायंस कि पेशेंट उसको ले भी उसमें कोई झिझक ना हो. सो यू सी हाउ मेनी डिफरेंट एक्सपर्टीज गेट इन्वॉल्व बियॉन्ड साइंस. कौन रेगुलेटरी उसका अप्रूवल मिलेगा या नहीं मिलेगा, एंड दैट इज व्हाट लीड्स टू दिस कॉम्प्लेक्सिटी दैट यू सी. सो आई विल कम बैक टू दिस कि ये सारा क्लिनिकल प्रीक्लिनिकल सारा काम हो गया. देन यू फाइल व्हाट इज नोन एज इन्वेस्टिगेशनल न्यू ड्रग. जब आपने ये सेटिस्फाई कर लिया पूरी तरह से कि ठीक है ये सारी प्री क्लिनिकल स्टडीज़ के अंदर, इन विट्रो इन विवो स्टडीज़ के अंदर जो आप देखना चाहते थे, यू गेट ऑल दैट सक्सेसफुली. देन यू गो टू रेगुलेटरी एजेंसी, जिस मर्जी कंट्री में, यूएस में फॉर एग्जांपल एफडीए, यू फाइल एक इन्वेस्टिगेशनल बिकॉज इट्स नॉट अ ड्रग येट, इट्स स्टिल अंडर स्टडी चल रही है, इन्वेस्टिगेशन उसका चल रहा है. इट्स कॉल IND, इन्वेस्टिगेशनल न्यू ड्रग एप्लीकेशन. उसमें आपको एवरी स्टडी यू हैव डन उसका डेटा इस लाइक इतना पूरा ग्रंथ भर के देना पड़ता है उनको. दे रिव्यू, दे आर सेटिस्फाइड कि ठीक है अब आप क्लिनिकल ट्रायल शुरू कर सकते हैं कि उसके क्या इफेक्ट है. सारे स्टडी वो दे आर ऑल साइंटिस्ट, दे आर हाईली ट्रेन हाईली एक्सपीरियंस साइंटिस्ट, दे आर सिटिंग एंड दैट्स व्हेन दीज क्लिनिकल ट्रायल स्टार्ट. सो इनिशियल क्लिनिकल ट्रायल होती है विद अ स्मॉल. सो ये फेज 1 लिखा है क्योंकि दैट्स अ ट्रेडिशनल. देयर इज अ न्यू प्रोसेस विच स्टार्टेड फेज 0 विच इज बेस्ड ऑन फार्माकोडायनामिक. सिर्फ 3 पेशेंट्स के ऊपर करते हैं स्टडी and they test the dose. If it is find effective, they change in the next 3 and the next 3. So between 6 to 9 patients, they have good idea whether the drug is going to be useful or not. Right? That's a PD driven, pharmacodynamic driven. But anyway, in general, there are 20 to 100 patients. This is purely only safety trial. कि drug safe है या नहीं. It's not the efficacy कि drug काम कर रहा है या नहीं. पहले सिर्फ safety कि वो safe है कि नहीं. Because if a drug is not safe, you don't want to give to more people, you stop right there. So again, go no go decision. Then you get into phase 2, larger population, and phase 3 which is worldwide. कि जो भी उसने कहा था intended उसका use है, उसका वाकई ही असर patients में नज़र आ रहा है. When there is a satisfactory and that is worldwide. And again उसमें भी बहुत सी चीज़ें हैं कि किस genes के लिए आप दे रहे हैं. आपका genetic makeup अगर वो नहीं है और तो उसमें उतने असर आते हैं कि नहीं आते. For example, we were approached when I used to be at the National Cancer Institute in US. We were approached and we were contemplating working with the government of India कि जो नॉर्थ ईस्ट के अंदर there is a genetic pool, उसके लिए ड्रग जो है स्पेसिफिक ड्रग्स तैयार करने के लिए, तो ट्रायल वहाँ करने के लिए, so that those patients can respond to that. So that is how, so phase 3 to, मतलब you try to pick your samples across the human race, different races के लिए, you know, चाहे black, white, Indian, Oriental, Chinese, Japanese, जो भी है, आप distribute करने की कोशिश करते हैं कि हर एक में वो काम कर रहा है. Then only it will go worldwide. So that becomes the largest and extremely expensive process. क्योंकि आपने उतने, आप just imagine एक ट्रायल के लिए, I just told you one IV करने जाना है, तो कितने क्लिनिशियन इन्वॉल्व है, कितने नर्सेज इन्वॉल्व है, कितने डॉक्टर्स, कितने बीच में जो है बिजनेस डेवलपमेंट पे, इट बिकम्स अ वेरी एक्सपेंसिव एंटरप्राइज. और वर्ल्ड वाइड अगर आपने करना है तो इस ह्यूज अरबों रुपए, इफ यू टॉक अबाउट रुपीज, और hundreds of millions of dollars to do that. So these decisions are taken very very carefully. But so again in terms of adverse effect, which is as I said in phase 1, सबसे पहले देखा जाता है कि उसके सेफ है, it is safe or not, before even go to phase 2. If it is highly toxic, you stop right there. So उसके safety pharmacology, chronic toxicology, idiosyncratic reaction, the reaction जिसके बारे में आपने सोचा नहीं तो अचानक पता नहीं कहाँ से साइड इफेक्ट आ जाता है। You study those things first. So and then you dose कि उसका डोज क्या होगा, क्योंकि जो भी आपने स्टडी की है इनिशियली that's all in animals. As the best case scenario, those are the studies you have data from the in vivo studies, animal studies, but now you are trying to bring it to the humans. तो ह्यूमंस में क्या डोज उसका होना चाहिए, कितने टाइम देना जाना चाहिए, उसको एडजस्ट करना. सो दोज आर द इनिशियल स्टडीज, इनिशियल क्लिनिकल ट्रायल्स की जाती है. किसी को आपने डोज दिया जो बेस्ट आपकी कैलकुलेशन जो है डेटा के अनुसार, प्री क्लिनिकल डेटा के अनुसार, इन विवो डेटा के अनुसार आपने दिया. अब तो कई फार्मूलाज बने हुए हैं कि यू कैन फार्मूला कि अगर माइस में है तो उसका ट्रांसलेट ह्यूमन में क्या होगा, पिग में है तो डॉग में है, मंकी में क्या होगा. बट दैट्स अ बेस्ट केस सिनारियो. जब आप एक्चुअली ह्यूमंस में लेके जाते हैं तो उसको थोड़ा एडजस्ट करना पड़ता है ऊपर नीचे. सो यू डू दोज काइंड ऑफ स्टडीज टू एडजस्ट कि उसको कितना देना है, कितनी बार देना है, उसका क्या डोज होगा. दोज आर द इनिशियल स्टडीज बिफोर दे आर इम्प्लीमेंटेड वर्ल्ड वाइड क्लिनिकल ट्रायल्स के लिए. सो उसके क्या इफेक्ट होंगे, यू सी फॉर एग्जाम्पल ये सारे टेस्ट करते चलते, यू कीप ऑन डूइंग ऑल दिस. डोज रेंज उसका क्या होगा, किस रेंज में देना चाहिए, एंड बेस्ड ऑन दोज डोजेज इज अगेन कि आपको पिल दिन में कितनी बार देना है या उसकी किस मतलब किस स्पीड से देना है, एंड हाउ व्हाट शुड बी. सो ऑल दीज नाउ यू कैन सी दीज आर ऑल क्लिनिकल स्टडीज, इनिशियल क्लिनिकल स्टडीज दैट टेक. एंड जब भी ये कर रहे हैं अगेन यू आर डिस्ट्रीब्यूशन, मेटाबॉलिज्म, काइनेटिक्स आपकी जो है, फार्माकोकाइनेटिक्स, ऑलवेज यू कीप दैट इन माइंड. ऑल ऑफ दीज आर बीइंग टेस्टेड नाउ इन द क्लिनिकल फेज में. ह्यूमन एनिमल्स में हो गया, नाउ इन द ह्यूमंस यू आर टेस्टिंग ऑल ऑफ दीज कि कोई जो है साइड इफेक्ट है, ऐसी चीजें नहीं है. क्लियरेंस क्या है, एलिमिनेशन क्या है, द सेम थिंग, बट नाउ इन द क्लिनिकल सेटिंग दीज थिंग्स आर टेस्टेड अगेन एंड अगेन. सो जस्ट टू गिव वन एग्जाम्पल, यहाँ पे अगर रीनल क्लियरेंस है, you try to see the compound की class क्या है. Again going back to chemistry, whether it is acidic, overall उसका pK क्या है, whether it's acidic or is it a neutral compound, आपका जो drug है, or is this on the basic side, उसके अनुसार उसके जो clearance है, अगर वो बनेगा तो ionized होगा या neutral रहेगा, polarity उसकी change होगी या नहीं होगी, because now that drug has to interact with various enzymes inside the body, inside the cell. तो उसकी प्रॉपर्टीज कैसे चेंज होगी, उसका क्लियरेंस कैसे होगा, इन सब चीज़ों को नाउ यू आर टेस्टिंग इन ए लेस नंबर ऑफ ह्यूमंस, बट यू आर टेस्टिंग ऑल ऑफ दीज थिंग्स साइमल्टेनियसली एंड क्विकली. सो दैट इज व्हाट यू डू. उसके बेसिस पे यू आइडेंटिफाई क्या है, एंड देन उसका क्लियरेंस कितनी जल्दी उसका हाफ लाइफ के अंदर क्लियर हो जाएगा, ताकि उसका बैठे बैठे रह के कोई साइड इफेक्ट नहीं होगा उसका. सो उसके मेटाबोलाइट्स क्या बनेंगे. Again, you can see each one here again, go no go decision, yes or no, आगे लेके जाना चाहिए या नहीं लेके जाना चाहिए. For example, in vitro और in vivo metabolism उसका करते हैं, structure activity relationship बनाया कि metabolite will likely to circulate. अगर हाँ जो metabolite बनाया it will circulate inside the human body, okay, then it's good, you take that drug forward. If the metabolite is not going to circulate, उसके stop, there is no point. You again go back to the blackboard, design the structure and keep. So this is, you keep on these yes no decision, and step by step you bring the drugs forward. Same thing, this is metabolite, same thing for pharmacokinetics. You look at क्या काम करता है in silico, in vitro. So now there are many techniques, many things are available. Oral लेना है, tissue में distribution क्या है. जब when we do in vivo studies for example, so if they are looking for let's say a type of cancer, so students in my lab and postdoc scientists they're working, they will do अपना intended use क्या है, उसके अलावा then they collect the other organs कि जो drug दिया उसका distribution दूसरे organs में क्या हुआ. they do pharmacokinetic study, उसमें से extract करते हैं कि हमारा drug वहाँ तक पहुँचा तो कितना पहुँचा या नहीं पहुँचा, तो आपका drug distribution क्या हुआ वो आपको पता होना चाहिए drug बनाने के. so when students do this or scientists do this, they collect आपका इंटेंडेड जो भी जिस चीज़ के लिए आप टारगेट कर रहे हैं वो कर रहे हैं, बट उसके अलावा भी आपका डिस्ट्रीब्यूशन क्या है बाकी सारे ऑर्गन्स के अंदर, यू कलेक्ट दोज ऑर्गन्स. सो व्हेन दे कंप्लीट देयर स्टडी, वट एवर लेंथ ऑफ स्टडी दैट दे आर डूइंग, they at the end collect these samples and then they do the pharmacokinetics, drug distribution क्या हुआ इसका. So all of these things are part of the whole process. So when you have, as you see here, so again you can now in silico इसलिए क्योंकि there is, as I was telling yesterday, big data, all that information so much is available that there are now such programs that are available which can at least guide you. They are not going to tell you what will happen in human, but इतना data available, they can just tell you कि क्या नहीं काम करेगा. So you don't waste your energy and time into it. So you can do in silico study. 10,000 कंपाउंड है, इन सिलिको देख के टॉक्स फार्मा एडमी के पूरे प्रोग्राम बने हुए हैं आजकल. देयर आर कंपनीज, पीपल डू दैट. वो बेस्ड ऑन द प्रायर नॉलेज, अगर कोई नया आप डिज़ाइन कर प्रायर नॉलेज के बेसिस के दे सकते हैं कि ओके दीज आर द फिजियोकेमिकल क्लासेस बेस्ड ऑन ऑल द एल्गोरिथम रिटन, इसमें से इतना काम करेगा, डिस्ट्रीब्यूट होगा नहीं होगा. सो इट नैरोज डाउन. आपके पास में 10,000 स्ट्रक्चर हैं, इट कैन नैरो डाउन कि भाई इनमें से जो 200 स्ट्रक्चर है इसी को आगे लेके जाओ. दे हैव पोटेंशियल, सो दे कैन सेव यू टाइम एंड मनी. So you can do in vivo, but eventually it is for human body. You have to do in vivo studies. You have to do systematic study, but it can save you time and frustration and resources. So when you do all of these things, bring it down to the human studies. So if you look for that, एक ह्यूमन डोज बनाने के लिए कितने स्टेप्स इन्वॉल्व हैं? जीनोटाइप, फिनोटाइप, किस जीन्स के लिए आप कर रहे हैं, उसका फिनोटाइपिक मेकअप क्या है जिसके लिए आप ड्रग कर रहे हैं. यू स्टार्ट फ्रॉम देयर, राइट. ड्रग की कॉन्सेंट्रेशन क्या है, किस टारगेट पे आप लेके जाना चाहते हैं, उसका प्रोटीन टारगेट क्या है, उसका जीन टारगेट क्या है, उसका बॉडी के अंदर टारगेट क्या है जिसके लिए आप ड्रग तैयार कर रहे हैं, उस टारगेट के अंदर उसकी फंक्शन क्या होगी, राइट. और उसका फिजियोलॉजिकल रिस्पॉन्स, बॉडी उस ड्रग के ऊपर क्या करेगी, एडमी टॉक्स क्या होगा, बॉडी उसको रखेगी या नहीं कि पैथोफिजियोलॉजिकल प्रोसेस हो, उसका डिस्ट्रीब्यूशन किस तरह से होगा सारा, और उसके बाद में उसका क्लिनिकल हाल में क्या होगा उसका. सो यू एज यू सी सिंपली फिजियोलॉजिकल रिस्पॉन्स लेने के लिए यू स्टार्ट आस्किंग दिस, नैरोइंग डाउन टू दिस मल्टीपल स्टेप्स टू गेट टू कि ह्यूमन एफिकेसी उसकी होगी या नहीं होगी, होगी तो क्या होगी. And that gives you the estimation of the therapeutic dose. उसके बेसिस पे therapeutic dose डिसाइड करते हैं, उसकी frequency क्या होगी इन patients के अंदर. Again, many a times, it's not most always, but many a times even the sexual or gender based के for female and for male, depending on what it is given, the doses are adjusted. In many cases, डिस्ट्रीब्यूशन एज ग्रुप का क्या है. आप देखिए जो पीडियाट्रिक्स जो हैं, बच्चों को जो डोज दिए जाते हैं, दे आर एट डिफरेंट लेवल देन द एडल्ट लेवल. दोज आर एडजस्टेड क्योंकि उसकी बॉडी उस स्टेज पे कितना रिस्पॉन्स करेगी, कितना करेगी या नहीं करेगी, कितनी उसकी क्षमता है, इट कैन एब्जॉर्ब, बायो अवेलेबिलिटी, ऑल दीज थिंग्स आर टेकन इनटू कंसिडरेशन. एनी क्वेश्चन? या COVID-19. Say it again. It was? The drug or the? No, I'm sorry. Could you be louder? So COVID-19, the vaccine was. आपका क्वेश्चन क्या था? I'm sorry, I missed it. It is effective in the short period of time. या नहीं? Oh हाँ. So what he's saying is कि जो COVID-19 की जो वैक्सीन है was brought into market in a very short period of time. The reason for उसके लिए था कि वो जो छोटा सा जो है उसने पूरी कायनात को बंद कर दिया था, सबके सर्वाइवल कर रहे थे, सारे रिसोर्सेज उसके पीछे लग गए थे. You know how much effort they put in. So you see, there were every country where they were trying to develop something कि इस enemy के साथ में tackle कैसे किया जाए. So many studies. If you think for a second, I showed you clinical trial. So this is my last slide for this. I showed you this clinical trial के कितने phases हैं. You have exploratory proof of concept, go no go decision, bring it to the phase 2, phase 3 and do. It could take from initial से लेके 10 साल लग जाते हैं कि ड्रग एक्चुअल मार्केट में ड्रग बने, अप्रूवल हो गया, इजीली 10 to 12 years it takes it. उस केस में क्या था? So you have to see for example, I mentioned phase 1 clinical trial up to 20 patient, up to 100 patient. Phase 2 trial 200 से 400 patient. Phase 3 clinical trial 200 से maybe 5-6,000 patient, right? कोविड के केस में तो क्लिनिकल ट्रायल बिलियंस में हो रही थी, जहां भी ड्रग जो डेवलप करने की कोशिश करता कहीं ना कहीं ट्राई कर रहा था डायरेक्टली टू बी पीपल का थिंक बस ड्रग स्टार्टिंग पॉइंट वाज कि ड्रग्स दोज वर देयर इज अ लॉट ऑफ knowledge that was already there. So coronavirus, why is it called corona? You know that? उसका जो है क्राउन, जो प्रोटीन का एक क्राउन सा बनता है. सो सरफेस प्रोटीन जो है, जब वायरस जो आता है वो सरफेस प्रोटीन पे आता है और उस सरफेस जो प्रोटीन है उसके साथ में इंटरेक्ट करता है और अंदर चला जाता है सेल के, एंड देन इट डिस्ट्रीब्यूटेड फास्टर. सो कोरोना क्राउन जो उसका है ना, सरफेस का जो सरफेस जो क्राउन है प्रोटीन का, उसका फॉर दिस वायरस इट हैज द प्रोटीन हैज अ क्राउन शेप. कोरोना इज द स्पेनिश वर्ड फॉर क्राउन. सो उसका सो कॉल्ड क्राउन है इंटरेक्ट्स with the surface proteins, it interacts and then it goes inside the cell और then फटाफट उसका वो जो है फैलना शुरू कर देता है. So the efforts were happening to stop that interaction उस surface proteins के साथ में. If you stop the effort of that protein के साथ में interaction जो है, जो proteins जो surface proteins है उसके बीच में जो इंटरेक्शन, सो ड्रग्स वर बीइंग डेवलप के जो इन दोनों का इंटरेक्शन है उसके बीच में उसको काट दिया जाए या उसको मिसलाइट किया जाए ड्रग को. सो ड्रग इज लुकिंग लाइक इज एंटरिंग इनटू द सरफेस प्रोटीन बट इट इज इंटरेक्टिंग विद समथिंग एल्स, ताकि वो सेल के अंदर ना जाए और फटाफट रेप्लिकेट ना करे तो इतना ज़्यादा से फैल जाए. दैट वाज द एफर्ट मेड. सो इतनी जल्दी इसलिए आया क्योंकि जस्ट थिंक फॉर अ सेकंड, एवरीवेयर एवरी कंपनी कंट्री जहाँ पे भी नॉलेज उस तरह की अवेलेबल थी, पीपल वर ट्राइंग टू सी ओके लेट्स अटैक दिस, लेट्स अटैक दिस. तो पूरा वर्ल्ड में एक ही ट्रायल हो रही थी. तो एक फार्मा कंपनी करेगी 20 पेशेंट लेगी, 100 पेशेंट लेगी. उस समय आप सोच के देखिए कि 8 बिलियन लोग हैं, 7 बिलियन लोग, जितने भी लोग हैं, जितने अफोर्ड कर सकते हैं, तो क्लिनिकल ट्रायल बिलियंस में हो रही थी. तो काम करता है या नहीं करता जल्दी पता चल रहा था. एंड दैट इज व्हाट वर फास्ट. एंड फ्रैंकली there was so much prior knowledge available and that knowledge helped to understand and quickly do faster, to identify something जो कि वैक्सीन जो है कि उसका रेप्लिकेशन को रोक सके. बेसिकली क्राउन जो है इसे सरफेस प्रोटीन जो है उसके साथ में इंटरेक्शन. So people were trying to, as I was telling, remember yesterday I showed you this, day before yesterday, जो सेल साइकिल है, S phase, G phase, M phase, G2 phase, right? तो वो उसी तरह जो है. So people were trying to, now whatever was available in their toolbox based on the prior knowledge, companies, I hope Gilead for example, I remember one of the company, initially they had so much information available from based on prior decades of studies. That's just one company. Just imagine hundreds and thousands of those companies. So everybody based on their knowledge of the protein, of the vaccine, of the जो भी है उस knowledge को use कर रहा था. And basically उस समय people forgot about all other diseases. कैंसर, न्यूरोडिजेशन, इन्फेक्शन, सब कुछ भूल गए. इन्फेक्शन में खैर दैट्स इन्फेक्शन, पर सब कुछ भूल गए. सारे एक ही ट्री में ट्रायल हो रही थी, वो थी कोविड-19 की ट्रायल हो रही थी. देयर वाज ओनली वन क्लिनिकल ट्रायल एट द टाइम एंड देयर वाज सो मच. इसलिए प्रायर नॉलेज, मल्टीपल पैरेलल एफर्ट अक्रॉस द वर्ल्ड एंड दैट हेल्प टू, क्यों नॉट ऑल ड्रग्स गॉट पास्ड. कुछ काम कर रहे थे, कुछ नहीं कर रहे, बट दैट वाज. एंड जो ड्रग लेके आया फॉर एग्जाम्पल काटिकरी को, जिसका ड्रग जो है फाइजर ने जो किया, she had been working on the RNA vaccine. Now again scientists किस तरह से होता है, she was working RNA vaccines पे 80s में, and people said wasting time. उसको कोई grants ना मिले, कुछ ना मिले, वो साइड में थी, वो करती रही. जब कायनात के ऊपर असर आया तो उसी का जो नॉलेज थी वही काम आई. You never know how the knowledge which is sitting, but she is very persistent. I know her personally, I have interacted with her. Very persistent. आखिरकार वही लेके आया. But again, it's a combination of the prior knowledge that was there. It's the combination of many trials happening simultaneously across the world. But ढूंढ फिर के बोला चलो ये काम करता है. जैसे इंडिया में हो रही थी इतनी ट्रायल, राइट? Indian companies were trying, Indian government was trying. वैक्सीन में India is one of the largest producer of vaccines. इतनी कंपनियां जो काम करती हैं. सेम वे यूरोप के अंदर था, यूके, यूएस, ऑस्ट्रेलिया, मैनी जापान, बहुत हर कोई कोशिश कर रहा था और प्रायर नॉलेज को लेके, और सबके पास में एक ही टारगेट था, कोविड-19. उस समय किसी को कैंसर, न्यूरो कुछ नहीं था क्योंकि उसका उस एनिमी का पता नहीं था कि क्या है. तो it came fast, yes, which is a good thing, but it was due to investment of many years, the knowledge of many years that brought it to that stage. So, any question? This is my last slide for the day. Any question? Yes. I'm sorry, किसमें इंजेक्ट करते हैं?
जो हम एनिमल्स के अंदर स्टडी करते हैं, रैट्स में, रैबिट्स में, तो हम वो स्पेसिफिक केसेस भी उनके अंदर पहले मतलब बताते हैं.
मॉडल क्रिएट किए जाते हैं. That's इसी होते जो अनफॉर्चुनेटली देयर इज नो अदर वे. अगर आपने एक किसी भी कैंसर टाइप को करना है तो पहले उसके अंदर वो सेल लाइन इंजेक्ट की जाती है, उसमें वो डेवलप किया जाता है मॉडल, फिर टेस्ट किया जाता है उसका.
जरूरी है कि जो मतलब कैंसर हमारे अंदर जैसे क्रिएट करती है वैसे उस एनिमल के अंदर.
वही मैं अभी कह रहा था ना कि कैंसर का इफेक्ट तो होगा, उसके अंदर आपका टेस्ट भी कर सकते हैं आपको, बट इवेंचुअली क्योंकि उसकी फिजियोलॉजी ऑफ दैट एनिमल इज डिफरेंट दैन द ह्यूमन फिजियोलॉजी, बट देयर आर नो अदर वेज. आप ह्यूमंस में तो सारे टेस्ट करना शुरू नहीं कर देंगे जब तक ड्रग आएगा नहीं. एज आई वाज टेलिंग यू, माइस के अंदर किया जाता है, नॉट कि माइस और ह्यूमन जो है मेकअप इज आइडेंटिकल, बट इट्स रिलेटिवली ईजियर एंड फास्टर टू डू दैट. सो अगर ना भी हो, थोड़ा फ्रैक्शन भी उसका 70-80% भी हो, तो आपको कुछ आइडिया तो हो जाता है कि वेदर इट विल वर्क और नॉट. दैट्स हाउ दिस होल प्रोसेस इज सेट अप. सो यस, उसके अंदर पहले वो डिजीज को बनाया जाता है, फिर उसमें टेस्ट किया जाता है. सो आप अगेन स्टैटिस्टिक्स गेट्स इन्वॉल्व, मैथमेटिक्स गेट्स इन्वॉल्व, कि आप मैथमेटिकली उसे करते हैं कि क्या स्टैटिस्टिकली सिग्निफिकेंट है. बायोस्टैटिस्टिक्स, इफ यू आर नॉट इंटरेस्टेड इन डूइंग एक्सपेरिमेंटल स्टडीज नाउ, लैब में जाके, बायोस्टैटिस्टिक्स इज अ वेरी हॉट फील्ड. कि समझ पाना कि स्टैटिस्टिकली जो आप स्टडी कर रहे हैं वो कितना ह्यूमन के अंदर सिग्निफिकेंस है या नहीं, इट्स अ ह्यूज फील्ड नाउ. कि आपने सैंपल ले लिया, आपने 1-2 में टेस्ट कर दिया, खुश हो गए जी, एक में किया, एक में काम हुआ, वाज इट रियल और वाज इट तुक्का, राइट? फिर आप सीधा उसको ह्यूमंस में लेंगे, वहां पे तो काम नहीं किया. सो स्टैटिस्टिकली एट ईच स्टेज व्हाट इज द नंबर दैट इज इंपॉर्टेंट, उसके पूरे फॉर्मूला है, पूरे कैलकुलेशन. सो व्हेन वी डू एनी स्टडी वी हैव बायोस्टैटिस्टिशियंस इन्वॉल्वड. दे टेल अस दैट फॉर विच, इफ यू हैव टू डू माय स्टडी फॉर एग्जांपल, हाउ मेनी, सो 7 माइस पर कोहोर्ट, इन द सेंस के 7 माइस जिसके अंदर आप कुछ नहीं देंगे, 7 माइस उसके अंदर आप एक टाइप का ड्रग देंगे, 7 माइस जिसमें डिफरेंट जीन लाइन है उसमें दूसरा देंगे, 7. इस तरह से 7 ही क्यों लें हम, क्योंकि उसके अंदर से कितने परसेंटेज में असर नजर आएगा, दैट इज रेलेवेंट टू क्लिनिकल इफेक्ट. ऑल ऑफ दीज थिंग्स आर टेकन इनटू कंसीडरेशन. सो दैट इज हाउ दीज थिंग्स आर डन. क्योंकि आपने लिया, 1-2 में किया, काम कर गया, बोलो जी बिल्कुल काम करता है, बट दैट से तुक्का भी हो सकता है. यू हैव टू हैव व्हाट इज द सिग्निफिकेंट. इफ देयर इज अ ड्रग ड्रग इंटरैक्शन, कितना कम से कम में होना चाहिए कि दैट शोज के ओके नाउ यू कैन रिलाय ऑन द रिजल्ट्स दैट यू सी. So all these checks and it's a very advanced industry now. It's a very advanced scientific field, but all these points are taken into consideration. Any other question? If not, this is my last slide for the day. Enjoy.
Thank you, sir, for the brilliant exposition. It's now time for lunch. We hope you enjoy the meal and be back by 2 p.m. sharp.
छुट्टी हो गई है सबकी, घर जाओ।
Welcome back everyone. Hope you had a refreshing lunch. We resume with the tutorial with Dr. Aman Bhalla to take us through asymmetric synthesis in drug design, methods and generation part 2.
Just a second. So, very good afternoon. So, we have studied about the generations in our previous lecture, that is generation 1 which is substrate control, generation 2 which is auxiliary control, generation 3 that is reagent control, and what is generation 4? Catalyst control. Okay. So, till now you know what are the methods and how you can prepare the pure chiral compounds. Now we have to go about one very important part, that is the basis of asymmetric synthesis. The important thing which you have to check in all these 4 generations, which we have already gave a glimpse of that in our previous lecture, that how this can be done. Now, if you have to check this synthesis with respect to chemical kinetics, and for any chemical reaction what we are generally talk about in terms of kinetic, that is a rate of reaction from substrate to product via some transition state or an intermediate. How fast you reach the transition state, how fast the transition state or the intermediate turn into your product, you check it with rate constant. Okay. So, that is a very simple reaction. If you see on your screen what we are writing, that from a substrate which is a prochiral, तो prochiral substrate क्या लिया था हमने? Any example of prochiral substrate? Cyclohexanol, a simple aromatic ring with carbonyl group, that is your prochiral. Now you are going to get 2 type of products, okay, either the R or S. Why we are saying either R or S? Because we are doing asymmetric synthesis. Our objective is to either get exclusively R or exclusively S. So, just see in terms of kinetic, we can say from substrate to product R or from substrate to product S. So, this conversion is calculated via a rate constant. So, you say it is either Kr or Ks. So, in terms of simple kinetics, we have to see the comparison. Okay. We have to see the comparison, how fast and transition state and intermediate can be converted into your desired enantiomer, whether it is R or whether it is S. So, how this rate is going to check? If you see a simple rate equation, you do not have to go about this rate equation, but only one terminology which I just wanted to take from it. Okay, I am not a physical chemist where I am going to tell you the all about this equation, but if I say give some evident support for particular formation of isomer or enantiomer, how we have to look it. So in a simple way, from substrate to product via rate constant, you are going to check. So if you check the rate constant equation, in all the parameters there is an important factor, that is change in energy, that is the activation energy. For example, your substrate is converted into product when that activation energy is reached, when you reach the transition or intermediate state, then you will get your product. So in whole this scenario, the difference which you have to create is with respect to activation energy. Okay. So if your change is 0, for example, say in a simple way, 5 kilojoule energy is required for both the enantiomer. You provide the 5 kilo, 50% you will get it. But if there is a difference in activation energy of one enantiomer and the other, then you can provide the energy for that. Okay. So if we are seeing this graph, greater the difference in energy, higher the selectivity, because you want to prepare enantio selective or enantio specific. Say in a layman language, we are not talking about as a scientist. We have to explain to some kid about this. We can say that if we have to make A, we have to give 5 kilojoule energy, and if we have to make a B, we have to give 3 kilojoule energy. Okay. Now if your industry or your system is providing you, or you are giving only 3 kilojoule energy, will you able to get A? Because A के लिए तो कितनी चाहिए थे? 5 kilojoule. So this is a simple concept, that if we want to create only one particular type of enantiomer, how we can deviate their transition state, how we can make one enantiomer transition state so high in energy that cannot be easily achieved, whereas the other enantiomer, which is our target one, we can minimize its energy so that it can be supplied and you will get your product. Okay. अभी ये मैंने एक्सप्लेन नहीं किया. फाइव आपने कर दी तो जो आपका सबस्ट्रेट है उसके चांसेस सबसे पहले जो 3 है वो रीच्ड हो गई, तो योर सबस्ट्रेट इज डायरेक्टली कन्वर्टिंग इनटू व्हाट 1. जब आपने 3 की एनर्जी रे, योर सबस्ट्रेट इज कंज्यूमिंग, यू आर नॉट एडिंग सबस्ट्रेट साइड बाय साइड, you have taken it in starting. Okay, you provide the 3. So if you have taken 100%, as the 3 barrier is reached, you are getting exclusively 1. Okay, so if you see in that graph, what I want to say, you see we said that 2 type of enantiomer, one is S and other is R. Okay, if you see we are writing prochiral adduct. Okay, यहाँ पे आपको ये देखना है, this is your prochiral substrate. Now see, there are 2 transition states, one is for S and one is for R. So that has been written transition state S and transition state R. If they are going to make as a 50-50, their transition state barrier with energy level should be same. Okay, So if you start from the prochiral adduct, now can you see in figure which enantiomer is in larger quantity? That is the R1. The S enantiomer is very little. Why? If you see the comparison, the change in energy with respect to R and S has been down with the presence of some chiral auxiliary reagent. Okay. So now in a very simple way, if I am saying you are getting 50% of both the enantiomer, that's mean both the enantiomer have the transition state with same energy level. Okay, you are providing that energy, both transition states have been reached, then your product start forming. But if somehow, what I am saying, if somehow you are able to differentiate, you are able to change the energy of these two transition states, then you have a chance to reach your enantio selectivity or enantio specificity. So how you can change that? For that you required some chiral auxiliary reagent. So that reagent is such a specific that out of the two transition state, it is helping to reduce the activation energy of one transition state, not the other. So whose activation energy comes down, the percentage of that enantiomer is going to enhance. Okay. So if we have to study of all this discussion, okay, तो हमारा फिर conclusive point क्या होगा? What we have to conclude from here? The important conclusion is what? Non-equivalence. तो इसको अगर आपने याद रखना है या माइंड में बिठाना है तो इज अ सिंपल वे विच आई गेव द एग्जांपल ऑफ इथेनॉल. इथेनॉल में कार्बन के पास भी हाइड्रोजन है, CH2 के भी है, ऑक्सीजन के भी हाइड्रोजन है, लेकिन जब आप इन 3 डिफरेंट टाइप ऑफ प्रोटॉन्स को मैग्नेटिक फील्ड में रखते हैं, वो तीनों ही अलग तरह से रेजोनेट कर रहे हैं, वो तीनों ही अलग तरह से कपल कर रहे हैं, तीनों ही अलग तरह से अपनी पीक रिप्रेजेंट कर रहे हैं. तो आपने क्या देखा उस मॉलिक्यूल में? ऑल्दो CH3 has 3 hydrogen which have the same environment, CH2 has 2 hydrogen which has the same environment, OH has 1 hydrogen which has the different environment. So the question is that what have to done to get enantio selectivity? The only thing is that to create the non-equivalence. Okay. So in this case, how this non-equivalence has been created? Just by differentiating the energy barriers of both enantiomers transition states. Clear? आपको clear हो गया है? Okay, you want to ask something? Okay. कौन सी एक्स्ट्रा एनर्जी? ओके, जो मॉलिक्यूल आपका बन गया है उसकी स्टेबिलिटी इतनी ज़्यादा है कि वो तो आपका लोअर एनर्जी लेवल पे ही आ गया है ना. ओके, आई पुट इन अ वेरी सिंपल रिएक्शन. व्हेन यू आर डूइंग अ रिएक्शन इन लेबोरेटरी, you are refluxing the material, like you took ethanol, you put your substrate and do the refluxing. The amount of energy which is required for your product to form after the formation is ethanol temperature comes directly to 0. It will take time. So if the solvent is present, your energy is trying to take that solvent in a evaporate form. Okay, but your product is stable in ethanol. That energy, until and unless you keep refluxing it, then it is going to melt your. Okay, so whenever a plan of this reaction is there, you are using a chiral auxiliary reagent, it should be kept in mind that this requirement should reach in such a way that your products remain stable in that. Otherwise, you have to immediately stop it. You have to distill out the solvent so that your product will remain it. Okay. So sometimes if some substrate is left, that energy can be used for rearranged product. Your yield will be low. But as soon as possible, you have to get rid of that extra. Whatever the extra is there, you have to take challenge of that and you have to get rid of extra. Okay. So now I am concluding of these two slides in a way that what exactly we have to see. So just see the last line, what we have written: for the reaction to be stereo differentiating, these diastereotopic phases must be non-equivalent. So सारी asymmetric synthesis में जितनी भी आप जनरेशन 1, 2, 3, 4 किसी भी तरह की सिंथेसिस कर लो, उसमें सिर्फ एक काम करना है. इफ यू चूज अ सबस्ट्रेट, if you choose a auxiliary, if you choose a reagent, or if you choose a catalyst, they all four will be capable of creating the non-equivalence. तो अब मुझे जरा बताओ, जनरेशन 1 में जाओ तो जो उसमें जो सबस्ट्रेट है वो नॉन इक्विवेलेंस कैसे क्रिएट कर रहा था? कैन यू रिमेंबर द एग्जांपल? इन आवर जनरेशन 1 वी हैव अ साइक्लोहेक्सेन. साइक्लोहेक्सेन के साथ आपके पास कायरल यूनिट थी. कायरल यूनिट में क्या लगा था? हाइड्रोजन बिलो द प्लेन, मीथाइल. सो डिफरेंशिएशन क्रिएट कर दी. नीचे जब आएगा ग्रुप तो उसको हाइड्रोजन मिल रहा है, ऊपर जो ग्रुप आएगा उसको क्या मिल रहा है? मीथाइल. सो यू differentiated the two phase for the incoming group. Now the incoming group will choose which one will be the better one to enter. Okay, so अब आप सोचो सारी सिंथेसिस का जो प्लान ऑफ एक्शन है वो क्या है? टू क्रिएट नॉन इक्विवेलेंस. क्योंकि अगर नॉन इक्विवेलेंस नहीं हुई फिर तो रेसमिक है, फिर तो 50-50 है. ओके, so since the transition state TSR and TS leading to R and S products are diastereomers, therefore they are non-equivalent. अब तो आप सोचो नॉन इक्विवेलेंट कैसे हुई क्रिएट? आपके पास कायरल एनेंटियोमर बन रहा है, आपने कायरल ऑक्सिलरी डाल दी. कायरल आपके पास सबस्ट्रेट था, कायरल ऑक्सिलरी आपने डाल दी तो दो कायरल सेंटर आ गए, 2 आ गए तो क्या बन गया? डायास्टीरियोमर्स. सो डायास्टीरियोमर कैन बी डिस्टिंग्विश. इनेंशियोमर नहीं होते थे. अगर नेचर ने इनेंशियोमर ही कर दिए होते तो आपको इस प्रॉब्लम से एसिमेट्रिक सिंथेसिस ही नहीं करनी पड़ती. अगर वो डिफरेंशिएट होते, फिजिकल एंड केमिकल प्रॉपर्टी भी अलग अलग दे देते तो आप ऐसे ही उनको इनेंशियोमर को रेसमिक मिक्सचर को यूज कर सकते थे. सो दे हैव बीन कन्वर्टेड इनटू अ diastereomers. So these transition states differ in energies. Okay, now in most of the stereo differentiation reaction, the difference in activation energy is 2 to 3 kilocalorie per mole. आप मैक्सिमम जो रिएक्शन है वो कार्बन कार्बन डबल बॉन्ड के और कार्बन ऑक्सीजन डबल बॉन्ड के एसिमेट्रिक सिंथेसिस में कर रहे हो. इन दैट केस फ्रॉम 1 enantiomer to 2nd enantiomer, अगर आप 2 to 3 किलो कैलोरी का डिफरेंस भी अगर कर सकते हो, योर स्टीरियो सेलेक्टिविटी और enantio selectivity कैन बी अचीव. एंड दिस इज वेरी मार्जिनल डिफरेंस. बहुत ज़्यादा नहीं है कि यू रिक्वायर समथिंग विच हैव टू क्रिएट अ बैरियर ऑफ 10 to 15 किलो जूल एनर्जी. यू रिक्वायर्ड ओनली 2 to 3 किलो कैलोरी पर मोल. Also in majority of asymmetric reaction, it is only transformation of an sp2 to sp3. कार्बन कार्बन डबल बॉन्ड आपका sp2 है, कार्बोनाइल sp2 है। अब उस पर जब आप एक ग्रुप लेके आ रहे हो तो कार्बन की वैलेंसी 3 से कहाँ कर रहे हो, 4 में, टेट्राहेड्रल, सो दैट इज योर sp3। तो चेंज जो है वो sp2 से sp3 है। ओके, बहुत बड़ा चेंज नहीं रिक्वायर्ड है इस केस में। सो दस इन दिस रिएक्शन इट इज ओनली द स्टीरियो डिफरेंशिएशन ऑफ टू फेसेस। व्हाट आर दैट टू फेसेस? अगर आप इस मॉलिक्यूल को देखो, हमने प्लेन में दिखाया है, रेड बॉक्स में आप देखो, कार्बन ए इट इज अ डबल बॉन्ड। ओके, सो अगर आप इसको प्लेन में देख रहे हो तो आपको सिर्फ दो फेस की वरी करनी है। कौन सा फेस? एक आपका फ्रंट फेस, दैट इज योर साइफ, और री। आपको सिर्फ ये दो फेस के ऊपर ध्यान रखना है, किस तरह से नॉन इक्विवेलेंस इनमें क्रिएट की जाए तो आपका हो सकता है। क्लियर? सो अगर अब आपसे पूछे कि एसिमेट्रिक सिंथेसिस अचीव करना चाहते हैं, इफ आफ्टर दिस क्लास एनी पर्सन कैन केम टू यू कि एसिमेट्रिक सिंथेसिस क्या होती है, इसको कैसे अचीव किया जा सकता है? सर कुछ नहीं है, इनएंशोमर सिर्फ फेस से ही डिफ्रेंशिएट होता है। तो अगर हम नॉन इक्विवेलेंस क्रिएट कर दें किस रिएक्शन में तो आपका इनएंशो सेलेक्टिव प्रोडक्ट बन जाएगा। दिस इज द ओनली रीजन दैट which you have to see। ये आगे मूव नहीं कर रहा। ये इसमें चल नहीं रहा, ये आगे कहाँ? ओके, so for the reaction to be stereo differentiating you require non-equivalence। how this non-equivalence can be created? ये आपने सुबह किया है। पहला पॉइंट क्या बता रहा है? The non-equivalence of phases may arise in three ways। What is that phase? Firstly, there may be one adjacent stereogenic center in the substrate which renders one phase more sterically encumbered than the other। क्या मतलब क्या है इसका? कि आपके सबस्ट्रेट में जिस फंक्शनैलिटी पे आप रिएक्शन परफॉर्म करना चाहते हो उसके एडजेसेंट में एक ऐसा सेंटर प्रेजेंट हो जो स्टेरिकली एक फेस को ब्लॉक करे, दूसरे फेस को ओपन करे। सो दिस इज आवर जनरेशन फर्स्ट। सेकेंड, in this case auxiliary also comes in this। second, the attacking species may contain a stereogenic center which cause it to interact with the substrate more favorably with one phase than the other। ओके, सबस्ट्रेट आपका बड़ा सिंपल है, लेकिन जो ग्रुप आ रहा है उसके पास ही कुछ ऐसा प्रेजेंट हो दैट इट ट्राई टू फिट ओनली फ्रॉम द one side, not from the other side। okay, in this way we can create the non-equivalence। third, neither the substrate nor the reagent on their own may be chiral, but the attack is mediated by chiral catalyst। okay, हमारे पास सबस्ट्रेट में कायरलिटी नहीं है, कोई ऑक्सिलरी आप अटैच नहीं कर पाए, रिएजेंट में भी कायरलिटी नहीं है, चलो catalyst must have something that able to distinguish the both the phases। so that to sum up the heart of asymmetric synthesis, it lies the non-equivalence of diastereotopic phases which subsequently brings in the difference in the energy of एक्टिवेशन। अब बड़ा सिंपल सा एक एग्जाम्पल, जस्ट ओनली वन एग्जाम्पल मैं आपको बताऊंगा, ठीक है? यहां से एक टर्मिनोलॉजी आती है डायास्टीरियोफेशियल सिलेक्शन और फेस ब्लॉकिंग। Okay, now in this reaction, okay, if I simply say can you visualize this carbonyl group CO? आपने इस रिएक्शन में क्या विजुलाइज करना है? In this reaction you have to say this is your X, this is your CO and this is your R group. Okay, ये आपने विजुलाइज करना है. Or I can say you visualize this phone as a CO group. Okay, ये आपका मैं आपको चलो ऐसे शो करता हूं, ये आपका कार्बन है. ओके, दिस इज योर कार्बन. अब आप इस पे किसी ग्रुप को लेकर आ रहे हो, ये ग्रुप यहां से भी आ सकता है, ये ग्रुप यहां से भी आ सकता है. अब कार्बन आपका क्या है? पॉजिटिव. कार्बन क्या है? पार्शियल पॉजिटिव. यू आर ब्रिंगिंग सम नेगेटिव ग्रुप टू इट. सो व्हेन दिस नेगेटिव ग्रुप इज कमिंग, यहां फ्रंट में कुछ नहीं है. क्या है? नेगेटिव ग्रुप इज कमिंग टू पार्शियल कार्बन पॉजिटिव, इट विल मेक अ बॉन्ड. बट इन दिस बैक साइड पे आप ये सोचो, ये ऐसे थ्रू मी मैंने एक ऑक्सिलरी प्लेस की है, ये मैंने एक एरोमेटिक रिंग यहां पे हैंग कर दी है इस फेस में. अब बताओ, एक्स नेगेटिव इज प्रेफर टू कम फ्रॉम दिस साइड, और इसको तो ये एरोमेटिक रिंग बीच में है, इट हैज अ डीलोकलाइजेशन ऑफ पाई इलेक्ट्रॉन्स. इसके पास नेगेटिव चार्ज है, आप यहां से नेगेटिव चार्ज का ही स्पीशीज लेके आ रहे हो, तो क्या होगा यहां पे? रिपल्शन. ठीक है, तो अब ये एक कार्बन था, मैंने इसके साथ एक ऐसा ग्रुप अटैच कर दिया जिसके पास एरोमेटिक रिंग है. अब ये एरोमेटिक रिंग हैंग हो रही है 3 स्पेस में. ओके, तो ये ऐसे हैंग होके इसके साथ ऐसे पीछे आ गई. ये आगे भी कर सकते हैं, ये आगे आ गई. तो आपका टैग किस पे होना है? पार्शियल कार्बन पॉजिटिव पे. तो फिर आपने काम क्या किया? आपने 2 फेस में से एक फेस की सिलेक्शन कर ली, दूसरे को ब्लॉक कर दिया. तो उसको बोलते हैं फेस ब्लॉकिंग और डायास्टीरियोफेशियल सिलेक्शन. अब वो डायाफेशियल सिलेक्शन क्या थी? इट्स अ वेरी सिंपल रिएक्शन दैट नेप्थोल मॉलिक्यूल होता है, एनी कॉम्प्लीमेंट्री ग्रुप. अब आप जनरेशन सेकंड में जाओ. जनरेशन सेकंड में आपने क्या किया था? साइक्लोहेक्सेनोन था, उसके ऊपर कोई कायरल यूनिट नहीं थी, आपने एक ऑक्सिलरी प्लेस की थी. उस ऑक्सिलरी का काम क्या था? वो 3 डिमेंशन स्पेस में ऐसे हैंग करे कि उसके जो नेगेटिव ग्रुप है वो एक साइड इकट्ठे हो जाएं. When they come together on one side, they will create an electron density. So the incoming group already is a negative one, it will repel. From the other side, it has an open space. So that is called phase blocking. So the question comes that in asymmetric synthesis, how will you create a non-equivalence? We are creating a non-equivalence in a way that with the reagent, okay, we create a such type of environment where one group is present, the other is open, so that the incoming group can easily enter. Okay, so according to you, now very simple, as a chemist इसका मुझे आंसर आपसे चाहिए. अगर हम कार्बोनाइल पे और कार्बन कार्बन डबल बॉन्ड पे किसी ग्रुप को इंट्रोड्यूस करना चाहते हैं, तो फेस ब्लॉकिंग के लिए हमें कौन से फंक्शनल ग्रुप सेलेक्ट करने चाहिए? विच मस्ट हैव सम इलेक्ट्रॉन डेंसिटी. फ्लोरीन ग्रुप हो, एरोमेटिक रिंग हो, ताकि जो इनकमिंग ग्रुप आ रहा है आपका क्या है? RMgX. तो R इज योर न्यूक्लियोफाइल, न्यूक्लियस लविंग. तो उस न्यूक्लियोफाइल के एंट्री के समय आपने वहीं पे एक नेगेटिव चार्ज की शील्ड रख दी तो क्या वो आगे कार्बन पॉजिटिव तक पहुंच पाएगा? ओके, इफ यू हैव this carbon positive or this nucleophile is coming towards it and in between you place a sheet of negative charge, will it come close to this carbon? That means what you have done, you blocked it. So what you can block, either the front face or back face, upper face or lower face. तो जब आपने face को block कर दिया, that is face blocking. अब ये होगी नेगेटिव टर्म. आपने एक रिएक्शन में किसी चीज से ब्लॉकेज की है, दैट इज योर फेस ब्लॉकिंग. इसको अगर हम पॉजिटिव में देखना चाहें तो क्या कहेंगे? पॉजिटिव में हमें क्या फायदा हो गया? एक ब्लॉक हो गया, दूसरा फ्रीली ओपन हो गया, तो वो हमारी सिलेक्शन होगी. दैट इज डायस स्टीरियोफेशियल सिलेक्शन. और दिस इज द सिमिलरिटी ऑफ नॉन इक्वलेंस. इक्वल है तो 50-50 है, तुमने बीच में एक शीट लगा दी, दैट इज नॉन इक्वलेंस. इधर शीट है, इधर शीट नहीं है, ग्रुप कैन कम इजी. तो ये जो ब्लॉकिंग करनी है आपने, वो ऐसे ग्रुप होने चाहिए विच डिपेंड्स अपॉन व्हाट टाइप ऑफ रिएक्शन यू आर ब्रिंगिंग. अगर आप न्यूक्लियोफाइल को लेकर आ रहे हो तो आपने वहीं पे नेगेटिव चार्ज की कर दी. अगर कहीं पे इलेक्ट्रोफाइल आ रहा है तो वहाँ पे आपने पॉजिटिव चार्ज की. सो दिस इज योर diastereofacial selection, or you can say it is a face blocking. बट जब आपने ये ब्लॉकिंग करनी है वो ऐसे ग्रुप से जो खुद पार्टिसिपेट नहीं करेंगे, वो तो जस्ट फेस को डिफ्रेंशिएट करेंगे. लेकिन उसके बाद क्या है, वंस योर टारगेट इज ओवर दे कैन बी रिमूव्ड इजिली. दिस इज सिमिलरिटी लाइक यू डन इन दी second generation. Okay, so we came again to the last slide, that which we are talking of this. In this we said 1 2 3 4. Okay, so if you have to select any enantiomer for the synthesis, now I can say that you are in any company and you want to prepare only 1 2 3 or 4, what will be in your mind? That if there are 3 possibility, you have to mark them, you have to close them, you have to only give a room for the one, whichever is the selection. Okay, so it is very easy. How it is easy? For example, if you selectively want that your group should come from the front face, then what to do? Block the back face. You place a substituent in a such a way so that the ring which is hanging, that should be on the back side. But if you want to get the attack from the back side, you have to plan a group which will be on the front side. So I am going to give tomorrow all the examples with regarding to this, what type of chemical reaction. We will do the asymmetric aldol, we will do the hydroboration, alkylation, hydroxylation, where the selective enantiomer has been prepared. Any question with regarding to this? I am going to show you tomorrow all examples. They all are chemical reactions. Okay, there are asymmetric sharpless epoxidation, जहाँ पे हम प्योर DET लेंगे तो कैसे एक ही फेस से ऑक्सीजन इपॉक्साइड बनाते हो ना, आइदर अबव द प्लेन एंड बिलो द प्लेन, वो कैसे 3 डायमेंशन में जाएगा, डीएल्साल्डर रिएक्शन में कैसे जाएगा, एल्काइलेशन में कैसे जाएगा, हाइड्रोबोरेशन में कैसे जाएगा. आई विल गिव यू टुमारो. These 2 lectures is only for chemical reactions. Okay.
Thank you once again, sir, for navigating us through the complexities of asymmetric drug synthesis with such clarity and depth. Let's take a short 5 minutes break and then we will move into the final session of the day.
वो दे दो मेरे को। हाँ 1 2 3 hello, am I audible to all of you guys over here? Hello, hello.
Hope you had a refreshing 5 minutes break. Now, for our concluding session of the day, I welcome Pallavi to give a lecture on chemosensors for iron and iron detection, the versatile role of iron.
कर दूं start? A very good afternoon to all of you. Myself Pallavi. So today I am going to deliver a lecture on chemosensors for ferrous and ferric detection, the versatile role of iron. So moving ahead, I would like to ask, can you guys or anyone think that can you live your life without metals? Anyone, if he or she is thinking that yes, they can stay without metals, anyone out of you can stay without metals? Yes or no? No. Yes, very good. Nobody can stay without metals. So one of them is iron. What is iron? Iron resembles the symbol of Fe. So with atomic ये ठीक है फिर, okay. So it's a transition metal, or we can say that it belongs to a group of elements which can show a various oxidation states and can form a coordination complexes. It is lustrous silvery grey metal and that plays a critical role in both Earth's geology and biology. As we see around ourselves that the phones, mobiles, infrastructure, buildings, everything around us is basically made up of metals, and iron play crucial role among these all. As iron is widely used in construction, manufacturing and as an essential element in biological system, such as if we talk about hemoglobin, iron is present in the ferrous form in our blood. So moving ahead, this is the basic chemistry of iron, here having atomic number of 26 with 55.8 atomic weight and high melting and boiling point, which makes it more ductile, or we can say that it has a more strength, so it can be used for the formation of bridges, buildings, etc. So now, okay, fine, we are having a iron, it is already inbuilt in our body. So from where we get in the nature? It is also present in nature. How will extract it from the nature? It is present in the natural mineral resources known as ores, from where we get iron. These are the four main ores of iron, basically hematite, magnetite, siderite and iron pyrite. Out of all four, two are very main, that is hematite and magnetite. They have a high abundance of iron in them. So moving ahead, moving to the chemical properties and history of iron. It has chemical properties, oxidation state, activity, etc., etc. Firstly, we are going to discuss about oxidation states. Iron is present in +1, +2 and rarely in oxidation state of 0, that is pure metal, and is present in +4, +6, and some of these two are the main complexes which basically present in +4 and +6. So we'll discuss one by one. So iron in a zero oxidation state. So iron is rarely present in zero oxidation state as compared to +2 and +3, that is ferrous and ferric. It is the examples of that are, this is basically found in metallic iron, or we can say that as a free iron as a form of nails, constructions, tools, industries having the oxidation number 0. Further example is FeCO5. Here it is a metallic compound with carbonyl is having a neutral, so therefore the charge is going to be 0. So here it is, iron is in a zero oxidation state. Now third example is ferrocene. Basically it's a ferrocene analog. Basically ferrocene is in +2 oxidation state because there we have a pentene rings up and down side. But here we are talking about benzene rings, so therefore its benzene have no charge, so therefore it's a neutral and it can act as a sandwich compound. Here we can say that iron exists in a zero oxidation state. Now what happens if I will remove the 2 electrons from iron? It will exist in a +2. The configuration will come out to be argon 3d6, and due to pair of 4 unpaired electrons, it is paramagnetic in nature. So, have you ever thought of that how iron hold inside the water molecules, how iron stay inside the water molecules? This was explained by VBT, that how bonding take place. VBT considers the open hybrid orbitals of metal with the lone pair of ligands. Now what happens if I talk about VBT? It occurs in outer, it has two options, outer d orbitals or inner d orbitals. Outer d orbitals, if I take the example of FeH2O6 2 positive, here central metal ion is having a 2 positive charge because H2 is a weak ligand and with a no charge, so iron is in a +2 oxidation charge. So what will be the hybridization? Here if you look at the image, if I talk about Fe2 positive, it is 3d6, that is 4 unpaired electrons, and now so what happens is that now H2 will donate its electron to the sp and d orbitals of iron, so leads to the formation of sp3d2, and the geometry will comes out to be octahedral. if now here ligand is weak field ligand, so therefore no pairing will take place. There are two type of ligands, weak field ligand and strong field ligand. Weak field ligand will do the less splitting, therefore no pairing will take place. In strong field ligand there is a large splitting and therefore pairing will take place. Now if I talk about higher enough d orbitals, here the example is FeCN6 4 negative. It is in the form of ferrous. Now the electrons will get paired because of the presence of CN bond, because CN is highly strong. So it will use the inner d orbitals of 3d, 4s and 4p. So leads to the formation of d2sp3, and geometry again remains the same, that is octahedral in nature. These are the two examples. They both are in octahedral geometry. So this is a tetraes. It is it is diamagnetic, and in ferric it is paramagnetic due to presence of unpaired electron. Now, firstly I had discussed about the oxidation state, that was very basic as we had studied it in the junior classes. Now I talk about the reactivity. When we were kids, we used to see that we purchase iron or nails, we used to see that there is rusting of irons. What happens? When iron interact with the water in the presence of oxygen, leads to formation of iron hydroxide, which further dehydrates slowly and leads to the formation of rust, that is Fe2O3.XH2O. Now how we can avoid the rusting? By doing paintings, by using alloying, that is adding the mixture of metal and non-metals, oiling, etc. Now acid. How will, if when iron reacts with any acid, we can take, here I am showing the example of hydrochloric acid, it can be sulfuric acid, it can be nitric acid, any other acid. So what happens if I take up iron powder in a test tube and add a hydrochloric acid, and leads to the formation of bubbling of gas, which shows the hydrogen gas has been released, and the salt formation will take place, which shows that it reacts with acid and leads to formation of that particular salt. Now complex formation, I had already taught you that what the basic of complex formation is. So magnetic properties. We all were fond of magnets when we were kids, that how they work. We have a great curiosity. The curiosity behind it was a chemistry, that is because of the ferromagnetic nature, because they all are unpaired. Iron is having unpaired electrons, due to which it has a magnetic moment and leads to attract the magnet, or we can say the metals. Now due to the versatile nature of iron, we used to say that it has a very versatile nature, because it can easily switch from one oxidation state to another oxidation state, from +2 to 3, 3 to 2. So it has been used in Fenton reaction for the creation of OH radical. What happens when ferrous reacts with H2O2? It leads to the formation of ferric, and ferric further reacts with H2O2 and leads to formation of ferrous. Here again regeneration of ferrous has taken place, and again we can form the ferric, which shows that iron is highly versatile. Now, what is the main mode of this reaction was? The generation of OH radical, which leads to break down the pollutants, dyes, or other insecticides, pesticides, etc. Now, there are various alloys of iron and the uses. Steel basically formed from iron plus carbon, stainless steel, iron plus chromium, cast iron, iron burned with a high content of carbon, wrought iron, pure iron with the slag, and alloys we usually use iron and other metals such as used in aircraft, tools and heavy machinery. Now, how it has been used in our body? We are having an inbuilt iron in our hemoglobin in the form of ferrous. What happened? Each hemoglobin molecule has four porphyrin rings, and each porphyrin ring is having a 4 nitrogen atoms attached and 1 protein chain, and one side is vacant, and oxygen will come and bind to that state and leads to formation of oxyhemoglobin. That means that our hemoglobin is binding towards the oxygen, and which helps to transfer the oxygen from lungs to the tissue, and again from tissues it will move to the further cell. Iron binds reversibly with oxygen, which shows that iron forms a bond with oxygen, but it doesn't form that strongly that it can't break the bond or it can't lose the oxygen easily. So it is very, we can say that versatile in this form also, that it can easily remove the oxygen and bind with the oxygen. Now myoglobin is basically known as for the storage for oxygen for quick release. Whenever we go to gym or running or dancing, we need more oxygen because our energy is getting drained out. So at that moment, Myoglobin helps to use that energy which is stored inside it, and also contain Fe2 positive in a heme group. These are the hemoglobin structure, 4 porphyrin rings, and here this one is the myoglobin image. Now what is the role of it is already explained. Now oxygen binding and release. What is cooperative binding? It tells us when the oxygen binds with the one porphyrin ring, the affinity for the other binding of oxygen will increase. This was explained by cooperative effect, and once that attached to second porphyrin ring, the other affinity will increase due to the changes in the globular chains of we can say the protein. So when the oxygen is attached to a hemoglobin, it is a relaxed form. When there is a decrease in oxygen, we can say that oxygen is not attaching to hemoglobin, it is in a tense form. Now Bohr effect. Bohr effect basically deals with the pH. Now in lungs are basically basic in nature. It always need, if in lungs we say hemoglobin attached to the oxygen, it always attaches to the oxygen in the basic condition. It will not attach with oxygen in the acidic condition. So in lungs the condition is it should be high pressure and high pH. That will lead to the binding of oxygen in the lungs. And if I'll talk about, for example, in why it doesn't why we need a myoglobin in the cells for the release of oxygen, because in cells we are having an acidic medium. How tissues react with oxygen leads to the formation of carbon dioxide, which is acidic in nature, and leads to decrease in the pH, and therefore their myoglobin will act, no hemoglobin will attach. Therefore that is the role of myoglobin, that it will help to release the oxygen into the cells. Now, okay, iron is present in hemoglobin and all. How iron is stored in our body? Who will transport the iron in our body? There are three main: ferritin, transferrin, and siderophores. Ferritin basically stores iron in ferric safely inside the cells, releases it when needed. Transferrin. Mostly we have iron in a ferrous form. It moves to the stomach, acidic condition, it converts into it gets oxidized, it converts to a ferric. From there, transferrin will become active, it will transfer the ferric to ferritin for storage. Siderophores, these are iron containing complexes and transport the ferric, helps in transfer of ferric ions. Now detection using the iron. One of the very important tests is Brown ring test, we all usually use in a graduation time. This test is not for the detection of iron, this is the test for the detection of nitrate, but here iron is used for the detection of nitrate. What happens, we take a test tube. For example, I am having a ligand, I want to check whether it is having a nitrate or not. What I will do, I will take the compound, I will add ferrous sulfate and H2SO4. What will happen? Ferrous will convert into ferric, nitrate ion will convert into NO, and that NO will react with the remaining ferrous and leads to formation of this compound FeH2O5NO whole 2 positive. This will lead to formation of how we'll come to know that this has formed. This will lead to formation of that brown color ring. This is the whole about Brown ring test. From there we can come to know that yes, nitrate ions are present. This happened because of by the using of iron. So now this is all we came to know, that yes, iron is present and all. Now why the detection of iron is important? Why we want to detect iron in future? Why we are making so much chemosensors for the detection of iron? As excess or deficiency, both are bad. Deficiency in a body can cause anemia. Excess can cause liver diseases, heart problems, diabetes, etc. According to World Health Organization, it should be less than 0.3 ppm per day. Now, this creates the need for the accurate, selective, and sensitive detection of ferrous and ferric ions. Now, the main question is, how can I detect iron? What are the easy methods? I don't want that much of labor work. I don't want to waste my time. I want cost effective. What are the main reasons by which what are the main instruments or we can use so it will be easy for me for the detection? So what should I do so I can easily detect the ferrous and ferric? The reason are UV and fluorescence. They are the main we can say instruments by which we can easily detect the other metal ions also. I am here giving the example of ferrous and ferric. Now why UV and fluorescence? These are simpler and less expensive. We can allow real-time analysis. Naked eye for simple optical sensors can detect in aqueous also. They are having a fast response. We shouldn't have to wait for a long time of period for 2 to 3 hours. They are so sudden. And the sensors can be functionalized with triazole, shift base, silanes, etc., tailored for copper 2 or iron, etc. Now here we are talking about shift bases, triazoles and silanes. Now what are shift bases? Shift bases are compounds containing an amine group or azomethane group formed by the condensation of amine with aldehyde. Contains lone pair at the nitrogen which can act as a donor atom. It has a various coordination, like nitrogen is having a lone pair, so it can easily give the electrons to the metal and can form a coordination bond. Can be designed to select it. We can make derivatives, or we can say we can design a shift base accordingly, then according to the need of our detection. So here it is written that shift base can be designed to selectively bind specific metals by modifying the functional groups or backbone. Some shift bases show they are having many properties like anti-cancer, antifungal, anti-inflammatory, etc. The one of the best part is that it is one pot synthesis under reflux or a room temperature, catalyst free, or a mild acid catalyst required sometimes, not every time. So what are the main properties why shift bases can act as a good chemosensors? The properties are presence of C double bond N, multidentate binding sites, tunable structure, conjugation and planarity, colorimetric and fluorescent behavior, strong chelation with ferrous or ferric or any other metal. Now here we are having a donor atom. Nitrogen will interact with the donor atom and form a coordination bond with the metal like ferrous and ferric. Now the properties, we are one by one explaining the property. Shift bases often contain additional donor atom OH-COH, enhancing the selectivity and stability of metal complexes. Each modification of ligand allows specific selectivity for specific metals like iron. And conjugation and planarity. Conjugation leads to the delocalization of electrons and inducing the UV visible and fluorescence response upon binding of the metal. Now, colorimetric and fluorescence behavior. Binding with metals causes shift in the absorbance. It can be any shift, increasing wavelength, decreasing batho, hypo, etc. And chelation, further high affinity for metal ion enables selective detection even in the presence of other metal ions. Now, this was the work done in our lab by our seniors, where the amine was reacted with aldehyde, leads to formation of azo-dyes. These are the particular conditions under which azo-dyes are formed, and further reacted with alkyne leads to formation of azide, further formation of alkyne, and further reacted with azide to formation of organosilane. Now, UV visible. When we find that our ligand under the UV visible, on adding the iron, ferrous iron, there was a shifting of a peak from 371 to 358, and a new peak at 311 has been generated, which shows that there is some sort of, we can say that some sort of relation or interaction between the metal has been happened. Now, moving ahead, triazole. Hello. Yeah. Now triazole. Triazole is a heterocyclic organic compound that contains a 5 with 3 nitrogen atoms and the 2 carbon atoms. Here, when the triazole was formed, the 2 isomers were formed, that is 1,4 and 1,5. With we can say that 2 forms were forming, but as when in the use of copper, there was a proper that it leads to the formation of 1,5, which we mainly use in our labs. So this is the experiment performed in a lab where with aldehyde leads to the formation of here alkyne.
अच्छा इधर भी देख के करनी है हेलो चल पड़ा so leads to the formation of Schiff bases and further reacted with azide and leads to the formation of organosilane which was further then we further use the UV visible and fluorescence. In UV visible, we came to know that on the interaction of ferric ion, there was a shifting of peak from 325 to 290. That is a bathochromic shift. And in under fluorescence, it has been observed that on addition of ferric ion, there was a quenching in the absorption peak. So NMR was done and recorded, which shows that a main peak appeared at 8.83 and a peak at 0.5 also showed that the formation of complex has taken place. Now metal was further binded by the nitrogen of triazole and nitrogen of Schiff bases, which was further proved by NMR also. Now what are organosilanes? Organosilanes are basically a bond between carbon and silicon. They are widely used in the detection of metal ions like iron when combined with metal binding ligands like triazole, Schiff bases or other donor containing functional groups. Now this was also this is the also experiment done in a lab where firstly alkyne was formed and further reacted by with amine of fluorine and leads to formation of Schiff base and further leads to formation of organosilane. Here we get the signal of on addition of iron into the ligand we got a signal which a new peak has been appeared at 363. A new peak has been appeared at 363 which shows that there has some sort of interaction between the metal and the ligand. Now, okay, we come to know about the metal is showing different peak or at a different wavelength. Now, how I'll come to know that how much sensitive or how much minimum to minimum amount of ligand should be required for the sensing. So for that, titrations were performed. From titrations, we conclude the LOD, and the LOD came out to be 0.0723 into 10 raised to the minus 8 molar, which shows that it is highly, we can say that highly sensitive towards the iron. Now selectivity. While performing the selectivity studies, that how much, even in the presence of other metal ions, how much our metal is selective or how much our ligand shows selectivity towards the only single metal. In here, anti-interference study proves that it is highly selective towards the ferrous ion. Now from NMR, proper shifting of the peak has been taken place from triazole moiety from 7.24 to 7.32 and the shifting of signal from 529 to 551 were also observed, which shows that or which proves the formation of compound with the metal complex. The ferric was bounded with the Schiff base of nitrogen, oxygen and nitrogen of triazole. Now moving to the conclusion, iron is worse ferrous and ferric oxidation states, enabling redox based reactions. It forms stable coordination complexes with donor ligands, which is key sensor design. Chemosensors for iron detection has a high affinity for nitrogen, oxygen donors, change in color and fluorescence. Ligands like Schiff base, triazole and organosilanes are widely used due to their selective binding, chromophoric or fluorophoric properties. These sensors allow for a rapid, sensitive and selective detection of ferrous and ferric even in the mixed environment. So these are the publication of our lab under the supervision of Professor Gurjasprit Singh for ferrous and ferric. You can go through these publications. These are the highly sensitive chemosensors. There are many more also. I am showing few of them. So in the end, I would like to say iron is just not a metal, is a backbone of civilization and it is built inside us. So it's play a major role whenever life or a chemistry goes, iron leads. Thank you so much.
Thank you, Ms. Pallavi. It was indeed a wonderful and insightful lecture. Now I would like to call Ms. Renuka for the next lecture. स्क्रीन पे मत देखना, चलेगा उधर देख के।
मैंने बैटरी चेंज नहीं किया, मैंने बोला था पक्का।
अब क्या बोल करना?
अब इसमें बात कर सकते हैं।
Good afternoon, everyone. I am Renuka, and today I am standing here in front of you all to present a presentation that is based on the leishmaniasis and the God complexes used in the therapeutics of leishmaniasis. So here is my title, that is the God complexes in therapeutics, and how we can use that God complexes in the treatment of leishmaniasis. So basically, what is leishmaniasis? In Hindi, we basically known it as a kala bukhar. I think you every person of here is heard about it. So it is a parasitic disease basically, and it is caused by the infection with leishmania parasite, which is spread by the bite of infected sandflies, and over 90 sandfly species are known to transmit the leishmania parasites. It is caused by protozoa parasite, which is transmitted by the bite of infected female phlebotomine sandflies. And the disease affect some of the world's population, and it is totally associated with the malnutrition, population displacement, poor housing, a weak immune system, and the lack of financial resources. As we move further, there are basically the three major forms of this disease. The first one is the visceral leishmaniasis, and it is basically known as the Kala Azar. It is fatal if left untreated over 95% of cases. It is characterized by irregular bouts of fever, and it was estimated that 50,000 to 90,000 new cases of visceral leishmaniasis are occur worldwide annually, and very few of them, that is about 25 to 45% of cases, are reported to the WHO, that is the World Health Organization. The next one is the cutaneous leishmaniasis, and it is the most common form which causes the skin lesions, mainly the ulcers, which might be painful or painless. And These can leave lifelong scars and cause serious disability or stigma. About 95% of this disease in these cases occur in the America, the Mediterranean basin, the Middle East and Central Asia. The last one is the mucocutaneous leishmaniasis. It leads to the total destruction of the mucous membrane. The mucous membrane is a very sensitive membrane which is present inside our throat and nose basically, and it is the moist layer on our skin. So basically the cases of mucocutaneous leishmaniasis are occur in the Bolivia, Brazil, Ethiopia and the Peru. So there are the modes of transmission. The sandfly is the most basic form of the transmission of the leishmania parasite. It is the primary mode, and the infected female sandfly is the primary mode of the transmission of leishmania. The next one is the zoonotic transmission. As the name suggests, the zoo, which is totally represented the animals. So when the leishmania disease is transmitted from animals such as the dogs to humans through the same sandfly vector, then it is known as the zoonotic transmission. The last one is the rare transmission, that sometimes it is very less common routes that the transmission caused through the blood transfusions, contaminated needles, vertical transmission from mother to child, and very rarely through sexual contact or laboratory accidents. Here is the life cycle of the sandfly. Firstly, as you show in the screen, that the adult female sandfly give the eggs, and about 30 days after, larva was formed, and the growth of larva was finally causes the development of pupae, and then it again forms the adult in about 40 to 45 days. So there are some geographical distributions, and there are some factors also which affect that geographical distribution. So the mainly cases which occur of this type of disease are found in Africa, Asia, Latin America, and visceral leishmaniasis is commonly observed in India, Bangladesh, Nepal, Sudan and Brazil. There are some factors that are the vector presence, climate, geographical barriers and human population density. So there are some symptoms and signs of these all type of leishmaniasis. So it totally depends on what type you have. So first is the cutaneous, and there are the symptoms of them. That is the skin sores, which are represented in the picture. They might be painful or painless. It was totally depend on the patient to patient and the immunity of that patient. The next one is the swollen lymph nodes, abdominal discomfort, night sweats and weight loss. The next are the visceral leishmaniasis, and there are the symptom of that. The basic symptom of the visceral form is the swollen abdomen and the swollen lymph nodes. Lymph nodes are basically the bean-shaped glands which perform a very vital role in our immune system. In this picture, a child is suffering from visceral leishmaniasis was shown, and the next one is the mucosal, as the name represents that the mucus. So mucous membrane, as I told you, that it is present basically in our throat, in our mouth and in our nose. So it totally affects these type of organs. So partial or total destruction of mucous membrane in nose, mouth and throat was observable in this type of leishmaniasis. There are some diagnosis and treatment. So in visceral leishmaniasis, diagnosis is made by combining clinical signs with parasitological or serological tests. Serological tests are basically performed on the serums to observe the specific disease against the specific parasite. So the treatment of leishmaniasis totally depend on several factors, which include the type of disease you have, and the contaminated pathologies, parasite species, and the last one is the geographical distribution and the geographical location. All patients diagnosed with visceral leishmaniasis require prompt or complete treatment. As we move further, there are some medications which are basically used to treat leishmaniasis. The first one is the liposomal amphotericin B, miltefosine, pentavalent antimonials, and many more. There are some therapies which are basically given by the patients, by the providers. The first one is the thermotherapy, as the name suggests, the thermo, which is totally represent the heat. So in this, provider applies the heat to our ulcer and the area around it. The next one is the cryotherapy, as its name suggests, with the cooling. So provider cools an ulcer and the area around it for about 10 to 30 seconds per treatment. And the last one is the laser therapy, which was done by the handheld devices. There are some conventional therapies which I told you, and there are some recent advances which also include the immunomodulation, drug repurposing, and the nanotechnology. We are here to discuss about the medications one by one. The first one is the liposomal amphotericin B, and here is the structure of that particular medication, that it also contains a hydrophilic region, which in which carboxylic acid is present in the terminal, and which is basically the reason behind the nature of hydrophilic. The next one is the hydrophobic, which contain the poly in hydrocarbon chain, and the at the terminal side we observe the amine group, that is the mycosamine group. So these all constituents make the liposomal amphotericin B, and due to this complex nature, it can interact with our body, as our body also contain the lipophilic or lipophobic. So the medications are totally based on that type of things, so that they can interact with our body in a very well manner. So there are some side effects of this disease, that is the fever, chills, headaches, irregular heartbeat, muscle cramps, and pains. So basically there is a thing which is very notable, that what is the target side of that particular medication. So that is the ergosterol. Ergosterol is basically the principal sterol which is found in the fungal cytoplasmic membrane, and it is the target side of the lymphotericin B. So the next medication is the miltefosine, which was sold under the trade name Impavido, and here is the structure of the miltefosine. It is basically look like the natural phospholipid, that it contains a hydrocarbon chain, that is the long chain which is present in the left side, and in the right side there is a trimethylamine group which is attached to a phosphate group. So it is also similar to the structure and the complexes and the layer lipid membranes present in our body. There are also some side effects, in which the Stevenson-Johnson syndrome is the most observable side effect of this disease, in which the mucous membrane of our body, that is from our throat and from our nose, they are totally so they destruct by the rashes, and these rashes are not so much curable, and it take a lot of time to be cured. The next medication is the paromomycin. So it is a sulfate, and it is the aminoglycosidate, which is belonging to a neomycin family. It is a broad spectrum antibiotic, and it is administered by intramuscular injection. The side effects are relatively limited, mainly the ototoxicity. Oto word is related to our ear, and when our ear got injured by taking some kind of medicine, then it is known as the ototoxicity. So these are some side effects, and the local pain of at the injection site is also observable. So in addition to the paromycin effect, mitochondrial membrane potential and modifies the amount of polar lipid in the membrane, perturbates the fluidity of the membrane, which leads to altered membrane permeability. Here is the structure of paromomycin. The next one is the pentamidine. It was available as the Pentam and the Lomidine in the markets. It is basically a second line treatment. If the first line treatment was not so much effective, then the second line treatment was given to a patient. So the pentamidine is reported to enter inside the kinetoplast via arginine and the polyamine transporter, and then affect the parasite DNA synthesis. So when the DNA synthesis was blocked, then ultimately the cell death of that parasite was done. So there are some metals which are used in this treatment. The first one is the antimony. So basically in antimony was found in +5 oxidation state, and the +3 oxidation state of antimony is very active, and when this active form was injected in the patient's body, then it interfere with the parasite's metabolism by inhibiting enzymes like tryptophan reductase and many more. Then it also disrupt the parasite's zinc finger proteins, which are involved in the DNA replication and repair. So there are the two models of the antimony. In the first one, it was reduced to the +3 oxidation state, and then it performed its function. But in the active SB +5 model, the SB +5 antimony in the +5 oxidation state is automatically active and form the complex with the DNA topoisomerase and many other enzymes, and ultimately the apoptosis was done. So there are some limitations that it is toxic. Routes of administrations are potentially intravenously and intramuscularly. The next one is the drug resistance, and these are some limitations of antimony. So there are some other metal based approaches like zinc and magnesium are also used in the anti-leishmania, and when a DNA metallo-intercalated, that is the platinum, showed a remarkable anti-leishmanial activity which causes complete growth inhibition of leishmania. And the next one is the peroxyvanadium compounds which show potent inhibitors of protein tyrosine phosphate and inducers of anti-leishmania effects. As we move further, there are the zinc sulfate was also used as anti-leishmania drug, and it can be administered orally and been used from last decades in a medicine and dermatology. In vitro sensitives of Almazer and the Leishmania tropica strains to zinc were reported to higher than those of pentavalent antimony. It was delivered intralasally with success in cutaneous leishmaniasis, and it could have immunomodulatory effect and which help in the wound healing, that the healing of the ulcers. As more recently it was administered orally to the Iraqi patients, and which are suffering from parasitologically, that confirmed the cutaneous leishmaniasis was cured. And the species which are not identified and is known are only Leishmania major and Leishmania tropica, which are present in that Iraq. It was suggested that systematic antimonial injections of cutaneous leishmaniasis treatment were better than zinc sulfate, but oral administration of zinc sulfate makes it cheaper, more convenient its consumption, and nearly close cure percentage to systematic meldumine antimoniate injections without any serious effects. So there are some clinically based gall based drugs which are used in as anti-leishmania. So in general, some metal drugs that have been used as the NTD, that is the neglected tropical disease, and researchers of bioinorganic scientific community are investigating the potential of organometallic complexes in the treatment of this disease. So there are some drugs which are formed from gold complexes, and the phosphine ligand was also used to make it more effective of the gold drugs. So phosphine ligand considered very important class of versatile ligands which interact with metal as sigma donor and pi acceptor. Many gold complexes have been of great biological interest, which are basically of large variety and structural diversity, which include the alkaline, nitrogen, phosphorus and sulfur. As we move further, why we need more drugs to treat leishmaniasis? Is the drugs available in market are not sufficient, or they are not working? So current antimony based drugs are expensive, have numerous side effects, and are becoming increasingly less effective due to drug resistance. So novel treatments are urgently needed to combat this parasitic infection, as existing therapies often face the limitations. So, what about triazoles? As we are studied that triazole have a very good impact in the biological studies. What about the triazole if we use them as an anti-leishmania drugs? So, it is basically a 5-membered heterocyclic system which consisting of 2 carbon atom and 3 nitrogen atom, and triazole was used as anti-inflammatory, antiviral, antifungal and many more. So, synthesis of 1,2,3 triazoles, as 1,2,3 triazole was basically used as the anti-leishmania drug. So, so there is the copper catalyzed azide, that is the alkyne cycloaddition. So in this reaction, when one side we take the alkyne, and on the other side the organic azide was taken, and then the 1,4 isomer was formed. So there are basically the two methods in which they can be formed. The first one is the Huygens approach, and the last one is the second one is the click chemistry, that is the copper based approach. So the Huygens approach suffers from slow reaction, high temperature, and it is not regioselective. So we have to form only one type of compound, that is the 1,4, that which is more regioselective. So the copper approach is more better than the Huygens approach. So there are some properties of 1,2,3 triazole which are stable under the thermal and acidic condition, which are generally insensitive to redox hydrolysis and enzymatic hydrolysis. This type of ring is basic aromatic hydrocycle with a pyrrole type and 2 pyrimidine type nitrogen atom. As we move further, the question arises that can triazole used as anti-leishmanial drug? Basically the salt derived from the imidazole or from the triazole are obtained as potent anti-leishmania drugs. In recent year, they have received a very significant interest due to their versatile chemical characteristics, such as they have good stability, distinct electronic properties and tunable reactivity. In addition to their impressive chemical application, they are ionic liquids, organic catalysis, polymer materials and metal ligands. On moving ahead, they are recently focused on the strategy of designing new triazolium salt and investigating their anti-leishmania effect. It was observed that these salts derived from the triazole exhibited higher anti-leishmania activity in intramacrophage amostigate and the promastigate of the different leishmania species. Amostigate and the promastigate are the two stages which are observed in the growth of the leishmania, and they are totally described about the differentiation in the locomotion, that the amostigate stages was stable and they cannot locomote, and the promastigates can do locomotion easily. So it is worth noting that the triazolium salt group is crucial for enhancing anti-leishmanial effect of molecule, as it demonstrates better performance than neutral triazole, generally featuring the hydroxyl group attached to the 4th position of the triazole ring, or introduction of the epoxide group to the 4th position of the triazole ring makes them more potent against leishmaniasis. So there are some complications which are basically observed in the treatment of leishmania. The first one is the bacterial infection. As I told you that the ulcers are observed in that kind of disease. So it is also a secondary infection. They can make ulcers more worse. They can allow bacteria to get deep into your body, which can lead to serious illness. The next one is the scarring. It can cause permanent damage to your skin, mouth and nose, including severe scarring. The next one is the relapse. Some people's symptoms go away with treatment only to come back later, or if any person having the immunosuppressed, then the chances of relapsing is more often. The next is the septal perforation and the collapse. The mucosal leishmaniasis can cause a hole in the tissue between the passage of your nose, that is the nose septum or the nasal septum. The structure of your nose may break down in that kind of leishmania. This can cause disfigurement, infection and breathing problems. The pneumonia is also more complicated thing which was observed in the leishmania. Here is the figure in which the lungs or the bronchiole or the alveoli of the healthy person was shown, and in the next as the below, the pneumonia patient was shown that the bronchioles were swollen and the air space filled with the fluid. The post-Kala Azar dermal leishmaniasis, that is the PKDL, is large bumpy rash on your face and other area of your skin. It can appear while you have visceral leishmaniasis or after you have recovered. It can last long and cause severe scarring. The last one is the hemophagocytosis. This is the life-threatening condition that causes the organ damage. At last we are here that how we can prevent the bite of sandfly so that we are not facing that kind of disease. So how can I prevent? So the best way to prevent all form of leishmania to avoid the sandfly bites, especially in the area of leishmania where it is common, ways to avoid bites include cover exposed skin with clothing including long pants, long sleeved shirts and socks. Wear EPA registered insect repellent. The most effective repellent usually contain the DEET, that is the N-N-diethylmetatoluidamine. So kill sandfly indoors by spraying living and sleeping areas with the insecticide. Sleep in a room with the window closed or with screens in the windows. Sleep under mosquito bed netting. Some netting is pre-treated with insect repellent for added protection. Sandflies are much smaller than mosquitoes, so netting and screens should have very small holes to keep flies out. Thank you.
Thank you, Ms. Rashmi. That was a very fascinating session. With that, we conclude day 3 of the GIAN course. We are grateful to all our speakers for their valuable time and to each participant for your active involvement and thoughtful questions throughout the day. We look forward to welcoming you all tomorrow for another day of learning, exploration and academic growth. Have a wonderful evening ahead.